Good afternoon, ladies and gentlemen. My name is Yu Kyung-ho, KAST Fellow and a professor of Seoul National University. We are now starting the 43rd online KAST International Symposium on New Horizons of Nanobiological Therapeutics. As an organizer, I'm very honored to have a chance to open this 43rd KAST International Symposium. First of all, my special thanks goes to the President Min-gu Han of Korean Academy of Science and Technology. He has given full support and attention for this symposium. I'm also grateful for the attendants and interested people, participants from the Korean Society of Pharmaceutical Sciences and Technology. I also appreciate all the invited speakers. This year, in COVID-19 era, nanomedicine has been more influential than ever. As you may have heard, most global frontrunners of COVID vaccine technology are using nanotechnology for delivery of COVID nucleic acid antigen. So in this field, we should be very proud of that nanomedicine can do something to change the world, to improve the global health. I'm also very proud of introducing the stellar speakers from all around the world. They are the leaders in this field and Controlled Release Society and Journal of Controlled Release. Today, they will share the current status and future directions of nanobiotechnology. You may be thrilled to hear their concept and their experiences. And I'm really confident this symposium will be insightful enough to open new horizons for all the attendants from the KAST and attendants from the Korean Society of Pharmaceutical Sciences and Technology. Please enjoy this symposium. Thank you. And today, Professor Min-gu Han, the president of the Korean Academy of Science and Technology, has come to honor this symposium. Please welcome the President Han.
Thank you, Dr. Oh. Good afternoon to distinguished speakers and participants joining from Australia, Japan, Korea, and Singapore. It's alphabetic order, just mean anything. And good morning to distinguished speakers and participants from Israel, Portugal, and United Kingdom. I'm very much pleased to welcome all of you to the 43rd KAST International Symposium entitled New Horizons of Nanobiological Therapeutics. Today's symposium is a part of a series of academic meetings held since 1999 by the Korean Academy of Science and Technologies. The KAST International Symposium aims to provide good opportunity to exchange valuable scientific achievement among the renowned scientists from all over the world. It is a pity that we cannot afford to meet in a person due to COVID-19. Of course, we may miss a glass of wine after face-to-face workshop, but I'm still thankful that we can continue our academic journey through online. Just remember that we spend so much time at the airport and all the transportation from the home to the convention site, but we just due to the thanks for these new technologies, we just do this kind of thing at your home and at your office. Thanks God. Today, my special gratitude goes to Professor Yu Kyung-ho for her superb effort in organizing this symposium. Thanks to Professor Oh, we are very much honored with the presence of nanobiology experts from Korea and 6 other countries. I hope today's symposium not only provides the exchange of valuable research findings, but also strengthens the network among the participants for the future collaboration. Let me close by wishing you delightful and stimulating workshop. Thank you very much.
Thank you, President Han, for very welcoming remarks. Now, I'd like to introduce the session chair of Symposium 1. Session 1 will be chaired by Professor Young No-byun. He is a professor of Seoul National University and also a KAST Fellow. He is leading the Laboratory of Cell and Macromolecular Delivery System. Please welcome Professor Young No-byun, the chair.
Thank you, Professor Oh. Hello, everyone. My name is Young No-byun from Seoul National University. In this first session, we have 4 speakers. And as a convenience for this virtual symposium, I will introduce the 4 speakers before the presentation. After 4 presentation, we'll have a live question and discussion sessions. So please feel free to let them in the chat box of both the conference website or YouTube. And then I will collect all the questions and I'll ask the question to the speakers instead of audience. And now I'll introduce the speakers. First speaker is Professor Hideyoshi Harashima, who is a professor in Faculty of Pharmaceutical Science in Hokkaido University. And he's associate editor of the Journal of Control Release and Cancer Science. And he's an executive editor of Advanced Drug Delivery Reviews. He will talk about multifunctional envelope type nanodevice for gene delivery. And second speaker is Ilba Rupenthal, who is a director of Buchanan Ocular Therapeutics Unit in the University of Auckland in New Zealand. And she is General Secretary of Control Release Society. And she'll talk about active drug targeting in ocular drug delivery. And third speaker is Professor Ki-dong Park from Aju University in Korea. He's a fellow of the Korea Academy of Science and Technology. And Professor Ki-dong Park is Vice President of Korean Society for Industrial and Engineering Chemistry, and also he is a Vice President of Korean Interventional Medical Device Society. And he will talk about hydrogel-based strategies to therapeutics delivery. And last speaker is Ian Yang, who is Executive Director in the Institute of Bioengineering and Biotechnology in Singapore. And she is Associate of Nanomedicine, Nanotechnology, Biology, and Medicine. And in 2016, she was elected to the American Institute for Medical and Biological Engineering College of Fellows. She'll talk about biodegradable cationic polycarbonates as effective vaccine adjuvant. Now we'll start the session. And the first speaker, Harashima Sensei, please.
Thank you, Chairman, for your kind introduction. It is my great honor to discuss the progress made in our laboratory in the 43rd online KAST International Symposium, New Horizon of Nanobiological Therapeutics. Although I'm not able to speak to you directly, I will do my best. The world market of medicine is increasing rapidly, 7% in average per year, and this growth is predicted to continue. As shown in this slide, the market in North America is the largest, more than 40% in the world, and is growing rapidly. China's market is the second, more than 10%, and is also growing rapidly. Japan is the third, however, the only one among developed countries with negative growth rate. Europe, including UK, Germany, France, Italy, and Spain, occupy 80% with moderate growth rate. BRICS, such as Brazil, Russia, and India, are also growing very rapidly. These data have provided strong stimulation to the Japanese government to encourage cooperation between pharmaceutical companies and universities to create new kinds of medicine. It is widely recognized that there was a paradigm shift in the field of drug discovery and development in the 21st century. As shown in this slide, the contribution of small molecules decreased and antibodies have emerged as a new type of medicine. Antibody medicines currently occupy 70% of the top 10 ranking in the world. Nucleic acid medicines and gene therapy are expected to be the next generation medicine. This slide shows clinically approved nucleic acid and gene therapy medicines that have been recently developed. Spinraza was approved in 2016 as an antisense drug. Kymriah was approved in 2017, known as CAR-T therapy, and is the first gene cell therapy using a retrovirus in the USA. Luxturna and Yescarta followed. In 2018, Onpattro was approved by the FDA as the first siRNA medicine using DDS, where dealing MC3 DMA, a pH-sensitive cationic lipid, was used as a component of a lipid nanoparticle. I think that the invention of MC3 represents a major breakthrough technology. In 2019, Zolgensma was approved as a gene therapy using AAV9 as a delivery system. AAV9 is administered intravenously and penetrates blood-brain barrier, enters into neurons and reaches the nucleus, where their genome is transcribed constantly. Therefore, one shot is insufficient to cure the disease. On the contrary, Spinraza needs to be administered every half year throughout the lifetime of the patient. Although these technologies are advancing rapidly, a serious problem has arisen. Opdivo is very famous in Japan since Professor Honjo at Kyoto University won the Nobel Prize by finding a new strategy for curing cancers known to be immune checkpoint inhibitors, and the PD-1 antibody was approved as an antibody medicine known as Opdivo in 2014 by the Ono Pharmaceutical Company. However, the cost of Opdivo is 35 million yen, making it very expensive. When Opdivo was extended from melanoma to lung cancer, the Japanese government reduced the cost to 10 million yen, which still seems to be very high. Owing to the Japanese health insurance system, we are now able to use Opdivo. However, the budget of the Japanese government might break. As shown in this slide, the cost of new types of medicines are very high. Among them, the cost of Zolgensma exceeds 200 million yen. Under such circumstances, it is highly expected that non-viral or artificial DDS with a reasonable cost should be developed to overtake these highly expensive medicines. A paradigm shift also occurred in the field of DDS. Controlled intracellular trafficking was a dream in 20th century. However, now it is possible as evidenced by the case of Onpattro and Zolgensma. As to selective targeting at the tissue level, Doxy was approved in 1995 by the FDA using passive targeting via the EPR effect. As to active targeting, not only the liver but also the spleen, lung, adipose tissue, and brain are now within our delivery area. Challenges to penetrate blood-brain barrier is currently a hot area in DDS. We have been developing a multifunctional envelope type nano device abbreviated as a MEND. MEND was inspired by mimicking an envelope type virus, which evolved in nature, and the system was optimized during millions of years. We have been developing MEND based on the concept of programmed packaging. It is important to have a program to overcome all barriers inside our body. It is important to design functional devices which realize our program. It is also important to have nanotechnology to assemble all devices into a nano-sized structure under normalized condition. The first success in our research was the RED-MEN, the surface of which is modified with a steroidated octaarginine MEN, containing a fusogenic lipid that enhances endosome escape. The transfection activities of luciferase activity by the optimized RED-MEN shown in orange bar was as high as those of adenovirus vectors shown in the green bar. This success encouraged us to move to in vivo delivery. When we use an RED-MEN in the in vivo conditions, the cationic surface charge needs to be shielded by PEG, polyethylene glycol. Otherwise, cationic nanoparticles aggregate in the blood circulation and their biodistribution cannot be controlled. By introducing PEG into a MEN, the circulation time in blood increases in proportion to the density of the PEG, as shown in the red squares, which means we are able to control their biodistribution with pegylation. However, the transfection activity decreased dramatically as shown in the bars. By introducing PEG, we lose transfection activity. We call this phenomenon the PEG dilemma. How to overcome the PEG dilemma is our task. The PEG coating is important when particles are in the blood circulation. However, once they penetrate blood vessels to the extracellular space in tumor tissues, the role of PEG is over, and a naked MEN can enter into tumor cells more efficiently. To achieve this, we introduced a cleavable PEG by inserting an enzymatically cleavable peptide sequence between the PEG and the lipid anchor as PEG peptide dope, abbreviated PPD-MEN. In 2006, the Nobel Prize was awarded to Fire and Mello for their work on RNA interference. After this finding, there was a shift from plasmid DNA delivery to siRNA delivery because regulational hurdles for plasmid DNA-based gene therapy are much higher than those of siRNA-based nucleic acid medicines. Therefore, we also shifted from plasmid DNA delivery to siRNA delivery, since siRNA delivery is much easier than that of plasmid DNA. In the case of plasmid DNA delivery, there are at least two barriers, namely endosome and nuclear membrane, while endosomal escape is the only barrier for siRNA inside the target cell. The leading researcher in siRNA delivery was Professor Peter Cullis at the University of British Columbia. His system was called Stabilized Nucleic Acid Lipid Particles, abbreviated as SNARP. Professor Cullis is 10 years older than me, and I'm trying to compete with him, but with a different strategy. A key concept for overcoming the PEG dilemma is to introduce a pH-sensitive cationic lipid. The first generation was D-lin DMA. It was not common to see papers on DDS in Nature. However, the activity of D-lin DMA was reported in Nature at an ED50 of 1 to 2 mg siRNA per kg, which was not surprising to us, since our system, Arlet-Gala-Men, also has a similar efficiency. However, in 2010, the second generation D-lin KC2 DMA was reported in Nature Biotechnology, and the ED50 was improved to 0.02 mg per kg, a 100-fold improvement in vivo. It seemed impossible for us to follow them with our strategy. Then we shifted our strategy to use a pH-sensitive cationic lipid, since there was no other way for us to reach him. An original pH-sensitive cationic lipid, YSK05, was designed and synthesized in our laboratory, which was made by Yusuke Sato, a graduate student under the guidance of Mamoru Hyodo, an assistant professor. Hyodo is a chemist who studied organic chemistry in the laboratory of Professor Noyori, who is famous as a Nobel Prize awardee. As shown in this slide, right lower panel, the YSK05 men improved the ED50 by more than 15-fold compared to the conventional men with an ED50 at 1 mg per kg. The second generation of YSK13 men showed an ED50 at 0.01 mg per kg, which is close to the ED50 of MC3, 0.005 mg per kg, which was invented by Arnailam. The mechanism for how the YSK men can silence gene expression in hepatocyte is shown in the left panel. Once introduced into blood circulation, the surface PEG is rapidly dissociated from the men, and ApoE binds to the naked men, which is recognized very efficiently by LDL receptors on the surface of hepatocytes. After internalization, YSK lipids are protonated in acidic endosomes and exert its fusion activity with endosomal membranes. The siRNA is released into the cytosol to exert its silencing effects. YSK05 was applied to cure hepatitis C in chimeric mice in collaboration with Dr. Kohara at the Tokyo Metropolitan Institute. The optimized siRNAs are encapsulated into the YSK05 MEND, and 1 mg per kg dose of siRNA was administered to mice 2 times. Chronic HCV liver was normalized and serum HCV decreased significantly for 2 weeks. The second generation of YSK13 MEN was applied to HBV mice. Since the YSK05 MEN failed to cure due to the development of toxicity, single injection of YSK13 MEN at a dose of 5 mg siRNA per kg was enough to cure HBV. We were challenged to prepare our own original pH-sensitive cationic lipid which could exceed the efficiency of MC3. To achieve this goal, it was essential to prepare a chemical library as Alnylam did. In our case, we changed not only the hydrophilic nitrogen derivatives, but also the hydrophobic lipid tail and the linker to generate a large library. The lead structure was YSK12C4, which exerted the highest silencing activity in vitro. However, in vivo applications were limited due to its high pKa. By making our original chemical library, we successfully identified CL4H6 with an ED50 at 0.0025 mg siRNA per kg, exceeding that of MC3 at 0.005 mg per kg. CL4H6 LNP is the third generation, and its silencing activity in the liver is 24-fold more efficient than the first generation YSK05 MEN and 400-fold more efficient than that of RH-GALA MEN. We recognize the power of structural optimization for improving silencing efficiency, which comes from not only endosomal escape, but also stability in the blood circulation, uptake efficiency by the target cells, etc. Let's look at the safety issue of CL4H6 LNP. 7 mg per kg is a high dose, 2800-fold of the ED50 of CL4H6 to induce toxicity, and YSK13 induced liver toxicity as shown in the left panel, while there was no toxicity for CL4H6 LNP for AST and ALT. In vivo fates were examined and long-time accumulation was observed for YSK05 and YSK13, while CL4H6 disappeared rapidly from both the liver and spleen. We introduced an ester bond in hydrophobic lipid conjugation, which could be broken enzymatically in liver and spleen, and had no toxicity. CL4H6 LNP was applied for messenger RNA delivery. Design of experiment was introduced for this optimization to minimize a number of animal experiments needed. The most efficient formulation was B13, which consisted of CL4H6 sphingomyelin cholesterol PEG equals 60 to 5 to 35 to 1.5 in molar ratio. LNPs were prepared using a microfluidics device that was designed by Professor Tokeshi at the School of Engineering, Hokkaido University. I will discuss microfluidics in more details later. The optimized LNP could deliver messenger RNA and transfect it efficiently in vitro and in vivo, better than those of MC3. Genome editing is an advanced technology that is more applicable for gene silencing or gene therapy compared to the previous techniques such as TALEN, zinc fingers. And we developed an LNP optimized for CRISPR-Cas ribonuclear protein. Care is needed regarding the experimental conditions of microfluidics so as to avoid inactivating RNP. By applying design of experiment, we were able to optimize LNP, and B6 was found to be the best for silencing in HeLa GFP cell. More than 95% knockdown was observed, which is tenfold more efficient than previously reported values. Base substitution by the co-delivery of single-strand DNA was also examined, and more than 20% change from eGFP to BFP was achieved in HEC GFP cells. This system can also be applicable for CPF1 RNP as shown in this slide. When we consider the clinical translation of our technologies, it is important to have a reliable system for scaling up LNP. In recent years, microfluidics was introduced to the DDS field, and microfluidics was found to be greatly superior to conventional ethanol dilution method. We have also been developing a nanopackaging system in collaboration with Professor Yoshinobu Baba at Nagoya University since 2006. Recently, we started collaboration with Professor Tokeshi at the School of Engineering, Hokkaido University, to design our original microfluidics device, which has certain advantage to the commercially available ones. Professor Tokeshi is an associate professor in the laboratory of Professor Baba at Nagoya University and was recently promoted to the rank of professor at Hokkaido University. The new microfluidics device that was invented is shown in this slide. As the last, I would like to express my appreciation to my collaborators as shown in this slide. Professor Yuma Yamada is a group leader of mitochondrial delivery, and Professor Takashi Nakamura is a group leader of nano-DTS for cancer immunotherapy. Unfortunately, I could not introduce their study due to the time limit today. Yusuke Sato is now an assistant professor and group leader of the design and synthesis of new functional lipids. Professor Ikrami Khalil is a group leader of in vivo gene delivery to the liver, spleen, lung, and adipose tissue, and he is now in Asuta University as professor. Professor Hyodo is now in Nagoya University. I am most appreciative of all of the hard-working students in our laboratory, as well as all the researchers who have been kind enough to collaborate with us as shown in the slide. Thank you for your kind attention.
Good afternoon, everyone. My name is Ilva Rubenthal, and I'm an associate professor and the director of the Buchanan Ocular Therapeutics Unit in the Department of Ophthalmology at the University of Auckland in New Zealand. And down here, I've just put a little picture of Auckland for you, just to show you all the icons of the city. So if you were to zoom in here, you would see a number of boats parked down there, which is why Auckland is also called the City of Sails. Surrounded by a lot of water, as you can see. One of the other icons of Auckland is the Sky Tower, which is right in the middle of the city. And then we also have the volcano cones. So around Auckland, there's 52 of those volcano cones, and they're very nice to go for a walk on. Now, the chimney you see here is next to the Auckland City Hospital, and this is also where the medical campus is, where I'm working. And today I'm going to talk to you about active drug targeting in ocular drug delivery. Being the director of the Buchanan Ocular Therapeutics Unit, obviously most of my research is in the ocular drug delivery area. Now, the Buchanan Ocular Therapeutics Unit was established with funding from the Buchanan Charitable Foundation. We've got those two lovely people over here, Dr. Trevor and Caroline Gray, that donated some money to the university, and also obviously supported by my head of department, Professor Charles McGee. It was launched in December 2013, so it's about 7 years old now, the unit. And the aims are to provide teaching and education of pre- and postgraduate ophthalmology, optometry, but also pharmacy and nursing students, and to develop and translate ocular therapeutic related scientific research into the clinical setting. I'm going to tell you a little bit more about this today. Now, my team in the unit, we've worked on a lot of stimuli responsive systems over the past years, so systems that are responsive to ultrasound or laser light, so either injecting a hydrogel that you can then cure with light, shining it through the cornea, or we were also working on some nanoparticles, mesoporous microparticles that had kind of a barrier arm on them that when you shine a light on, you know, those barrier arms would open and would release the drug. We're also currently still working, and this work was started by Navid, on an electrical stimulus related implant that would release the drug when you put a small electric current on it. We're also doing a lot of work on new drugs, especially inflammasome blockers, and I'm going to tell you a little bit more about that, to treat inflammatory conditions in the eye, such as diabetic retinopathy, age-related macular degeneration, or uveitis. And we also work with new drug delivery vehicles. So Priyanka, for example, was working with semifluorinated alkanes during her PhD, technology that has been developed by Novalik. Yosra is working with a collagen-based hydrogel that's cross-linked with zinc oxide nanoparticles. And Henry has just started his work on extracellular vesicles for ocular drug delivery. If you're interested in any of our research, please have a look at our website. So the first example I wanted to talk to you about today is a novel fusion peptide for specific retinal targeting. And this work was done by Fraser, who's one of my former PhD students. Now, just to start off with, age-related macular degeneration, for those of you who don't know, so the macula is located at the back of the eye. Now, if there is fluid accumulation in that area, obviously whatever picture gets transferred, that gets blurry. So in a normal eye or with a normal macula, this is what you would see. If you have choroidal neovascularization, it gets all blurry. So what happens there on a, let's say, more cellular level? So if you ever look at the cross section of the retina here, and this is a schematic that's kept rather simple. We've got the choroid with a lot of blood supply, the retinal pigment epithelium, and then we've got the photoreceptors that obviously are very important for your vision, transporting the visual information. Now in AMD, if you have inflammation and ischemia in the choroid, what happens there is that those blood vessels become leaky and they're actually not able to supply sufficient oxygen to the retinal pigment epithelium, which results in hypoxia and ultimately vascular endothelial growth factor overexpression. Now, when that happens, you obviously get these new vessels forming to try and deliver more oxygen to the retinal pigment epithelium, but because they are leaky, they still can't give enough oxygen to the RPE cells, and eventually those RPE cells die and they can no longer support the photoreceptors, and this is how your vision gets lost. And then you get further neovascularization into the subretinal space and bleed out of those vessels, which results in fluid accumulation. Now, what is generally done in clinics is frequent injections of these anti-VEGF agents to try and prevent this overgrowth. However, it's not the ideal therapeutic. What we really want to achieve, because they're just kind of reducing those new vessels from forming, but they're not really fixing the problem, which is here. So what we are trying to achieve is vascular normalization, so actually repair those vessels here, and therefore maintain the integrity of the blood retinal barrier by using XG19. Now, what is XG19? Well, it's obviously an abbreviation. But before I get into what it is, I'll just have to go a little bit into the biology about the drugs that we're working with. So we're working a lot with connexin 43 hemichannels. Now, connexin 43 is expressed in most cells in the body. When two hemichannels come together, they form gap junctions, and this is how cells communicate. And the hemichannels themselves are usually closed. However, in disease conditions, so in inflammations, those hemichannels might open, and you have exchange with the extracellular environment, and this is how you get ionic imbalances with the cells, which can ultimately lead to cell death. Especially what gets released here is ATP, and I'm going to show you how that reacts on the inflammasome pathway on the next slide. So to prevent this from happening, we can block those hemichannels by using mimetic peptides. So there's a number of mimetic peptides out there binding either to the extracellular loop of one of those connexin 43 proteins or to the intracellular loop. And the intracellular loop is where our centric GAP19 is working. So, you can appreciate that usually GAP19 would have quite a difficult job to penetrate inside the cell. So we coupled it to a cell-penetrating peptide called Sentry, and it's a 5-amino acid cell-penetrating peptide. The reason I put peptide 5 is up here because the second example I'm going to give you today is about peptide 5. Now, so what actually happens with Sentry GAP19 or peptide 5? So when we block those hemichannels, those connexin 43 hemichannels, what we prevent is ATP from getting released out of the cell, binding to its own receptor and activating the NLRP3 inflammasome. So having this inflammasome complex forming, which results in caspase-1 activation and ultimately release of pro-inflammatory cytokines. So by blocking the connexin 43 hemichannels, we're really working right at the beginning of the inflammatory cascade. So what is Sentry? What does it bind to? We wrote an article last year on drug delivery if you're a little bit more interested in the technology. But Sentry binds to synakin 4, which is a cell surface receptor that is expressed in normal cells. However, it is upregulated in cells in injury. So whether it's hypoxia or, if you put cells into high glucose and inflammatory conditions, you get more of the cell surface receptors expressed. Meaning that if you, covalently link your peptide, for example, to Sentry, or if you complex your siRNA with Sentry, or you might even have Sentry on the surface of your nanoparticles, you're able to get more of your drug inside the cell, specifically into injured cells rather than normal cells. So does that actually work? Well, just to orientate you again, this is the schematic I showed you before, the cross-section of the retina. And now we're actually looking at a human tissue section, and we're looking at where that syndicin 4 receptor is expressed. So you can see here around the choroidal blood vessel, which is our target, as well as in the retinal pigment epithelium, which is also our target. So that's great. That's where we want the drug to be taken up and have its action. So if we're working with retinal epithelial cells, there's quite a lot of syndicin 4 already expressed in normal cells. However, when we then put them into hypoxic media, those syndicin 4 receptors upregulated. Now, does that translate into higher drug uptake? It certainly does. So under normal conditions, so these are just RPE cells with the cell nuclei stained in blue. And then we're looking at the uptake of XG19 here in yellow. So you can see here under normal conditions, yes, there is some uptake. But if we then put the cells into hypoxic media, the uptake is just massive. You get a lot more of the drug inside the cell. So that is good. So from then on, we thought, okay, can we actually see this also translating into animal models? So we have a laser-induced CNV in vivo mouse model where we're using pigmented mice. And what we are actually looking at is the optical coherence tomography images. So that's pretty much a cross-section of the retina to look at how thick these layers are or whether any of the barriers are compromised when you do the laser burns. And we also look at the fundus image, so pretty much just the surface map of the back of the eye. So what we do there, on day 0, we create laser burns around the optic disc, which is in the middle here, to rupture Bruch's membrane. And you can also see that here ruptured. And then we look at things like the formation of butterfly lesion on day 1 after the procedure, as well as the CNV lesion formation. So pretty much what I showed you on the cartoon here before, this is what's happening here on day 7. And we also looked at the size of those lesions over time. Now we gave our drug on day 0 straight after doing the laser burns. So what we could see is that looking just at the 2D fundus images and measuring kind of the lesion area, we could see a trend towards reduced lesion area with our XG19. However, that was not significantly different. But you can see here that it's actually quite difficult to map out where the edge of those lesions is. And you also can't see this way how deep the lesion is and whether there's any fluid accumulation. So then looking at it, in the OCT images and measuring the CNV lesion volume, we then found a significant reduction in the CNV ellipsoid volume when we treated those animals with XG19. And I should say the treatment was given intraperitoneal, and one advantage is that syndicin 4 is not expressed on circulating blood cells, which means that it doesn't get sequestered by the systemic circulation. So another advantage of this drug in terms of getting to where you want it without being taken up by cells that don't express syndicin 4. So this was the first example I wanted to show you. The second example is the work done by Dr. Di Huang, who's currently a postdoc at Harvard University. And she was working on hyaluronic acid-coated nanoparticles for cell-specific peptide delivery. So just to give you an overview of her PhD, so she was working with a mimetic peptide as well, but in this case, peptide 5, which works on the extracellular loop. With a peptide, you get poor stability, and they don't penetrate the barriers very well. So she decided to package them into human serum albumin particles to sustain the release and improve the stability, and then coat them with hyaluronic acid to achieve active retinal targeting. So hyaluronic acid binds to CD44 receptors, and again, it's been found that those receptors are regulated in disease. In addition to just using the nanoparticles, and I'm not going to show you any data on this today though, she also investigated the use of ultrasound to see if she can increase the mobility of those particles in the vitreous, which is kind of a very jelly substance, and whether she can enhance penetration by creating little pores in the retina to get the drug to where it needs to go. With the ultimate aim being, again, to get the drug after intravitreal injection to the RPE and the choroid. So Dee investigated a number of different parameters. So the first one was whether the peptide was adsorbed to the outside of the HSA nanoparticles or incorporated. And then, they were coated with hyaluronic acid. She then looked at the physical chemical properties of the nanoparticles. So your particle size, zeta potential, entrapment efficiency, looked at the morphology using TEM, so you can see that light gray coating around the HSA nanoparticles, and then looked at the in vitro drug release. So not looking at the data in detail here, but if we look at the bottom line here, which is pretty much the peptide adsorbed to the outside of the particle, the red line here, which is the peptide adsorbed to the particle, relatively high burst release, whereas if you incorporate the peptide inside the particle and then coat with hyaluronic acid, that is the blue line down here. So you get sustained release for at least 2 to 3 months. She then also looked at active targeting, so were the particles taken up by the retinal cells that express CD44? Was there any toxicity? Is the peptide still functioning? And also looked at ex vivo penetration through the retina. But since I will also have some in vivo penetration data, I'm going to focus on that on the next slide. So here we didn't use the laser CNV model, but a retinal ischemia reperfusion model. So how that model works is that, we've got our rats in this case, and we cannulate the anterior chamber, so just poke a needle just behind the cornea. And that needle is attached to a syringe and is attached to a saline bag that you put up on the wall to create a certain pressure in the eye, to create your ischemia. And then after 1 hour, you remove the needle and you get reperfusion. Now what you can see here, so we're looking on the left-hand side here, we're looking at nanoparticle uptake and distribution, and the particles are labeled with red. So you can see here that in an injured eye, you get a lot more of the particles penetrating to the area where you want it. Obviously, in an injured eye, barriers are compromised, and you do therefore get better penetration, which is great because that's what we're hoping where we deliver the drug to. So you can see a pretty big difference in a nanoparticle distribution after 24 hours. However, after 7 days, we found pretty much the same amount of particles, whether the eye was injured or it wasn't injured. To our surprise, and not in line with what we've heard or read in literature, is that we didn't find CD44 upregulated in our retinal ischemia reperfusion model. However, we just looked at cross sections, and in the future, what we'd like to do is really do retinal whole mounts to be able to see small changes in CD44 labeling in the eye. So just looking at the right side here, just to then quantify where those particles were in the different layers. So hardly anything obviously goes to the cornea when you inject something into the vitreous. Surprisingly, not much left in the vitreous, but probably because of the circulation that happens in the eye. A lot in the retina and a little bit of it in the choroid sclera. Now, does that translate into therapeutic effect? So the first thing that Dee did was looking at the diameter of the arteries and the veins at the back of the eye. So we've got the veins here on top and the arteries at the bottom. I'm not going to go into too much detail here, but black is our control animals that did not, that had not any retinal ischemia reperfusion done. But you can see here that when animals, they were injured and they only had saline injected, that the diameter of those vessels significantly drops over time because you get bleed out of those vessels and they shrink, whether it's the veins or the arteries. However, if you're trying to normalize those vessels, working with our connexin 43 hemichannels, you do get some recovery. Not as much with the free peptide, so in red here, but a lot more with the peptide incorporated into nanoparticles. And that trend is similar when we're looking at the retinal layers. So here we're looking at the OCT images. So again, a cross-section of the retina, looking at the different layers and the thickness of them. And again, I'm not going to go into all of the data, but let's maybe concentrate on this figure C here, where again, we have the change in the thickness of the inner nuclear layer, which is this layer up here. So in the injured animals that only got a saline injection, you can see how this layer thins over the period of 8 weeks. However, if you give the particles, the nanoparticles with the connexin 43 mimetic peptide incorporated, you're pretty much back to baseline levels. And last but not least, we're also looking at immunohistochemistry. Obviously, our protein of interest, which is connexin 43 in green here. So again, we can see there's a little bit of green in obviously our control animals. Once you injure the animals, you get some more connexin in the retinal ganglion cell layer and in the RPE layer. And when you then treat, this is again reduced, as you can see here. So increased with injury. With the nanoparticles, we're almost back to baseline. We also tried a mixture of the particles and the free peptide just to see whether we need some peptide straight away to have an action straight away. So, same concentration. So this was 20 micromole, this was 20 micromole, and here we did 10 plus 10. But you can see here that the particles by themselves probably, more sustained release and obviously stayed around for longer, had a better effect than combining the two. The other marker we're usually looking at is GFAP, which is a marker of inflammation. So again, you can see here a lot of red streaks going down, massive upregulation of inflammation when you injure the animals but don't give any drug, and then reduce when we're giving the drug, again, back to baseline when we're having the drug incorporated into the nanoparticles. So with that, I'd obviously like to thank my team for all of the amazing work they're doing, especially Dee and Fraser, whose work I presented today, all of our funders, and obviously Caroline and Trevor for establishing the unit and supporting the unit, and all of our collaborators, and thank you for your attention.
Good afternoon, everyone. It is my great honor and pleasure to be part of this wonderful symposium. I would like to thank KAST for organizing this international symposium, especially Dr. Han Min-gu, President of KAST, and Professor Oh Yu-kyung of Seoul National University for this wonderful opportunity. Today, I would like to introduce our hydrogel-based strategies to deliver therapeutics. I would like to start my presentation by expressing thanks to our lab members and the collaborators. And one more announcement before I start my scientific talk. I'm the organizing World Biometric Congress 2024 to be held in Daegu City, Korea. I would like to invite all of you to participate this World Biometric Congress. We are expecting 5,000 participants, and we'll make a wonderful scientific program in addition to the social program. Okay, let me start my talk. Main platform today, I would like to introduce the injectable and sprayable hydrogel. To make injectable and sprayable, we have to make in situ forming a hydrogel. This can be utilized as therapeutic scaffold and therapeutic vehicles to deliver various biologically active molecules. In situ forming hydrogel, they provide easy to use and high functioning scaffold, and we can expect a minimally invasive implantation procedure, and we can make easy and homogeneous cooperation of cells and bioactive molecules. And in addition to these major advantage, we can expect many unique characteristics by using this kind of injectable hydrozols. So to make this kind of injectable in situ forming hydrogel, we can deliver a drug, or we can deliver cells to make a sustained drug delivery system, or expecting enhanced tissue regeneration and augmentation. So today I would like to various application model that we have tried by using various platform injectable hydrogels. I would like to say how to make injectable hydrogel systems. As you know very well, there are two different scenarios. One is we can make a chemically cross-linking system or a physically cross-linking system. So we can utilize a variety of chemical cross-linking reactions as shown in this slide. Nowadays, many scientists are utilizing click chemistry, and if we think about chemical hydrogel, we can enhance mechanical stability, but we have to always think about, we have to concern a toxic potential due to the additives. Meanwhile, if we make a physically cross-linked hydrogel, this is a very simple way to make in situ forming hydrogel, but there's some limitation in a mechanical strength and stability. So over last 10 years, our laboratory has focused on a new system utilizing enzymatic reaction, so-called enzymatic cross-linking system. If we utilize the enzymatic reaction, as shown in here, peroxidase and peroxide chemistry, this is very simple way. I will talk about this one a little more. And this enzymatic cross-linking system has been reported 2002 by Professor David Kaplan, Tufts University, first time. And we utilize this system too. So, as shown in this slide, if phenol derivatized polymer are cross-linked under peroxidase and hydrogen peroxide, we can get an in situ forming a hydrogel due to the carbon-carbon new bond formation or carbon-oxygen new bond formation. So this is a kind of very simple way to make a quick gelation. So if we utilize this phenol derivatized system, we can apply a variety of matrices. For example, we have reported PEG-based block copolymer, tetronic, prolonic, or multi-arm PEG, or glycerol sabacate. These are synthetic polymer, and natural polymer, including gelatin, keratin. Keratin is human-based protein, chitosan, hyaluronic acid, heparin, arginate, etc. So if backbone are derivatized with phenol group, we can make in situ forming hydrogel by using this system. And I would like to add this comment. This in situ cross-linkable hydrogel by enzymatic reaction, we can control their physicochemical property easily. For example, gelation time, we can control from seconds to minutes or longer than that, and we can control mechanical property. In addition, we have advantages if we utilize peptide protein, which has a tyrosine residue. We can easily conjugate into the backbone polymer. Just a one-shot process. When you make in situ forming hydrogel, we can conjugate peptide. And usually, a polymer backbone that that I have mentioned are biocompatible, and we can make it a biodegradable or a bioabsorbable or bioerodible. So chemistry is very simple. We can apply a phenol derivatized system from synthetic polymer to a polysaccharide, protein, peptide, etc., etc. And how can you control gelation time? Just simply changing the enzyme concentration. In the case of mechanical property, how can you control the modulus value? Yeah, just changing hydrogen peroxide. People are worrying about the use of toxic hydrogen peroxide, but less than 10 years, we optimized, and our backbone is biocompatible, non-toxic. And if we increase the hydrogen peroxide concentration, definitely cross-linking density increases, but above a certain concentration, definitely concentration exists to deactivate the activity of enzyme, so decrease the cross-linking densities. Okay, so by using this peroxidase peroxide, or instead of hydrogen peroxide, we can utilize glucose oxidase or calcium peroxide, which system we can have hydrogen peroxide generation system. So automatic hydrogen peroxide are generated. This generated hydrogen peroxide can be utilized for the cross-linking reaction. In addition, we can utilize this small dose, high dose of hydrogen peroxide for therapeutics. So I will introduce this system, and hydrogen peroxide, or if we make precursor derivatized system, we can make our hydrogel to release the nitric oxide. Or if we utilize calcium peroxide, then resultant oxygen generation, we can utilize the oxygen generating hydrogel system too. And we can make ROS convincing system. This is very important when we deliver stem cell, etc., etc. So let me start this therapeutic application. Okay, this slide summarizes the therapeutic applications that we have tried, but due to the time limit, I want to introduce some application. First, let me start a functional peptide delivery system. As I mentioned, okay, this one shows the therapeutic delivery. Mainly, our works to deliver therapeutics focus on tissue regeneration, tissue wound healing system, organ regeneration, etc., etc. So as you can see in this slide, cardiac tissue regeneration, or some bone regeneration, or parathyroid hormone delivery by using stem cell, skin ulcer, wound healing. Yeah, we reported several papers by delivering dendritic cell and virus, oncolytic virus, as a cancer immunotherapy, but muscle, nerve regeneration, etc., etc. So, as I mentioned, due to the time limit, I'll move on to peptide delivery. So my collaborator, our collaborator, Professor Park in SNU Dental School, they discovered calcium accumulating peptide, so-called osteogenic peptide. So this CAP has tyrosine residue in there, so we conjugated this CAP peptide into backbone matrix. So we compared the free releasing system or chemically conjugated system. We compared two systems, how to induce osteogenic differentiation of mesenchymal stem cell and bone regeneration. So if we utilize conjugated system, low rate of release, and if we just without tyrosine residue Y, we wrote CP here. CP quickly release out. So in vitro osteogenic differentiation study demonstrated that CAP has definitely better than CP. So slow releasing by conjugated system, this one demonstrated better results. And these in vitro results are consistent with in vivo results by red calvaria model, as you can see here. So CAP conjugated injectable hydrogel system demonstrated highest bone volume and bone density value. Okay, this is one peptide delivery system. Second one, I will introduce a growth factor delivery. So if you think about just the hydrogel, we first strategy is to change the cross-linking density. So modulus will be different. So high cross-link density hydrogel shows low swelling data, and if we increase the if we decrease the cross-linking density, high swelling. So by swelling ratio, we can control the protein delivery. In here, we can utilize this one, demonstrate with NGF and FGF and the sequencer delivery system. And the second one is if we utilize nanogel. In here, we utilize the heparin polonic nanogel, which has been studied for various purposes. And we combine this heparin polonic nanogel together with the hydrogel system. So we can manipulate tiny control of release rate. So as you can see in this slide, if we deliver FGF loaded into a hydrogel, macrogel, and release rate can be very fast. And if we combine with nanogel, we can deliver at the sustained slow manner, as you can see right bottom. So to deliver long-term growth factor, so we can utilize this heparin polonic nanogel together with this hydrogel system. So I will show you one delivery effect of dual growth factor. As I mentioned, neuro NGF and FGF, these are very important growth factor for tissue regeneration. So we utilize this one for erectile disorder disease. So heparin polonic bind FGF due to the heparin affinity, and NGF was just embedded into a hydrogel. So we can expect faster release of NGF and slow release of FGF. So we studied this dual sequencer release technology, and we measured in vitro and in vivo function. So erectile function analysis, as you can see here, in a normal and dual delivery using heparin polonic and heparin polonic with NGF and heparin polonic FGF. So demonstrated erectile function it's close to the normal. And the other single growth factor delivery demonstrated not enough functional recovery. And the muscle relaxation, if we see the muscle relaxation data, we can see a similar results. And some other biological assay, NOS, ENOS, etc., and small smooth muscle actin and apoptosis analysis support this dual system is more efficient compared to the single growth factor system or control. This is a dual growth factor delivery using heparin polonic nanogel together, heparin polonic nanogel embedded hydrogel system. Second one is growth factor plus stem cell delivery system. This one was delivered by using in situ forming hydrogel, our hydrogel, and we utilized in vivo lacerated mouse model to evaluate the muscle regeneration. And the data shows compared to the single growth factor delivery and the stem cell delivery, this growth factor and the stem cell dual delivery system demonstrated better, less fibrosis formation, and contractility study demonstrated, functional recovery and some new blood vessel formation and nerve regeneration, and this kind of functional recovery, revascularization, reinnervation for muscle regeneration have been achieved by using this stem cell, fat-derived stem cell, and growth factor delivery system. Next example is stem cell, by using a stem cell delivery to deliver parathyroid hormone. So as you can see in this slide, thyroid surgery causes a permanent loss of parathyroid gland function. This one lead to the no ability to control of calcium level. So our collaborator in Ewha Women's University, Professor Joe, they have a unique tonsil-derived stem cell. So we utilize this stem cell, and this stem cell generate hormone. But if we inject this stem cell, burst release of a hormone is expected. So we try to deliver a controlled manner of parathyroid hormone by delivering tonsil-derived MSC using our injectable hydrogel system. As shown in here, cells are viable. This C-TMSC, D-TMSC, C is a control, D is a derivatized stem cell. So all viable for 2 weeks, and their hormone release behavior, differentiated tonsil-derived stem cell shows a better release. So we compare this control stem cell and derivatized tonsil-derived stem cell and our hydrogel and matrigel. As you can see here, so our hydrogel delivering system demonstrated highest in vivo survival even compared to the matrigel utilized system. And I skipped this one due to the time, and also stem cell delivery for cardiac tissue regeneration. Same way, we can utilize our injectable hydrogel for cardiac tissue regeneration. I do not explain in more detail. Okay. And I would like to show several examples by delivering low concentration of ROS, high concentration of ROS. So low dose of hydrogen peroxide is very effective for angiogenic activity. We know the hydrogen peroxide is toxic. If we utilize a high dose of hydrogen peroxide, it's toxic. But we can utilize this one for antibacterial coating or antibacterial activity. So our hydrogel can generate ROS concentration, ROS at different concentration. So if we control the small dose of hydrogen peroxide, we can expect our matrix to be angiogenic, new blood vessel formation. So this is very important technology for tissue regeneration. So we manipulate hydrogen release kinetic under 10 micromole for 2 days, as shown in this release kinetic, and they show enhanced endothelial cell activity and intracellular RS level was upregulated. And we are doing an in vivo study, but in our study, we confirmed the enhanced angiogenic effect by delivering small, low dose of hydrogen peroxide. So we can utilize that. And if we utilize this one, we can deliver different concentration of nitric oxide too. So nitric oxide precursor, SNO, can be conjugated into backbone matrices. So we can make hydrogel in situ, and this hydrogel deliver nitric oxide. I do not want to explain in more detail. So ROS, RNS, so depending on releasing amount, so we can utilize for different purposes. Previous speaker demonstrated excellent system for ocular delivery. I would like to introduce our hydrogel system for ocular delivery, because previous speaker introduced very well for many eye disease, especially AMD. We can deliver Lucentis and Avastin. That's the current therapeutic. So there are many ways in an intravitreal injection. There are many ways. So we tried this injectable hydrogel system using hyaluronic acid microgel and to inject vitreal body, and we confirmed the 3 months pharmacokinetic. And I didn't show safety data in here, but it's biocompatible. So we are optimized this 3 months of pharmacokinetic system. In addition to here, we would like to enhance the long-term delivery. So as I introduced to you, this kind of in situ forming hydrogel are very useful for therapeutic implant and therapeutic carriers for various therapeutic agents. Thank you very much for your kind attention.
Very good afternoon, everyone. Before I give my presentation, I would like to thank Professor Yu Kyung-oh for her very kind invitation. And also I would like to thank Professor Byung for chairing this session. It is my great pleasure and also great honor to be here to share our research, especially in the area of biodegradable cationic polymers as effective vaccine adjuvant in this prestigious symposium. Vaccine adjuvants have been used to stimulate an immune response against cancer or infection through T-cell mediated lysis of infected cells or cancer cells. It can also induce the production of antibodies to enhance body immunity. various types of adjuvants have been reported over the last few decades, and these adjuvants include the aluminum particles and also oil in water emulsion and water in oil emulsion, even virusomes and others as well. Among all these adjuvants, aluminum particles have been widely used in clinic because it is low cost and also can actually induce immunoresponse for various antigens. And there are limitations and because of its usage. And the limitations include that the localized inflammation, injection site pain, and tenderness and intensification of IgE antibody response, which is an indication for allergic reactions. And also, more importantly, in certain diseases like HIV and influenza and also the dengue, and the aluminum particles were not effective to induce immunoresponse. And this is because it is unable to amplify the T cell mediated immune response, which is important in these diseases. As such, we aim to develop polymers as adjuvants that are biodegradable, cost-effective, and safe to use without causing harmful side effects. And it is inspiring that the polymeric nanoparticles formed from mannose modified the polyethylene imide, the short form, the PI, are really promising. They are currently undergoing the phase 3 clinical trials. So in our study, we have designed and also synthesized cationic polymers, and we use these cationic polymers to complex with antigen, like the model antigen that we use is OVA, and also to complex with poly-IC, and which is also used as an immunostimulant. And we actually, the polymer can complex these two actually components into nanoparticles. And then we use these nanoparticles to vaccinate mice and see whether the mice vaccinated can actually against cancer challenging. And here I would like to explain that the poly-IC is a synthetic analog of double-stranded RNA, and it is really a good immunostimulant. It can generate a strong Th1 responses, and also it is ideal for the anti-tumor vaccination and also for vaccination against pathogens which requires a T cell component such as HIV, influenza, and dengue infection. So we synthesized two types of polymers, and basically these polymers are diblock polymers and containing a cationic block of polycarbonate and a hydrophobic block of polylactide. And these two polymers have different component of polycarbonate and polylactide, especially one is with a shorter chain of polycarbonate and shorter chain of polylactide. And another one have longer chain of cationic polycarbonate and longer chain of polylactide. And the reason for us to use cationic polymers because it was reported and cationic polymers can induce a stronger immunity compared with neutral or anionic materials. So the polymer was synthesized by using a really advanced chemistry so that we can control each block length very well, so we can do a comparison and study the effect of different composition on the vaccination efficacy. So these two polymers were used to complex with the OVA, and from these two tables, we see that the two polymers can complex OVA into nanoparticles with narrow size distribution at a lower mole ratio of a polymer to OVA. However, at a higher mole ratio of polymer to OVA, it formed the particles with multiple peaks in terms of size. And as we see from the PDI, here it has a wide size distribution. and especially compared to the polymer with the longer chain. And this polymer can have the particles with the lower cationic charge surface. And it is expected that the particles with a lower cationic charged surface will have a lower cytotoxicity in vitro and also in vivo. Therefore, in the further study, we have used this polymer for the complexation of a poly-IC. So we see from these two tables, and especially for the top table, and the polymer can complex poly-IC into very fine particles with narrow size distribution when the mass ratio of the polymer to poly-IC at 0.5 or 2.5. And we see here also at 2.5, we have positively charged surface. However, at 0.5, we have negative charged surface. And also when we actually loaded the OVA and poly-IC into the same particles, and it formed really large particle aggregates at a mole ratio of 8.8 to 1, and 16 to 1, and 32 to 1. The size ranged from 4 to 6 micrometer. And we also can see, and the zeta potential here is much actually around the neutral, and especially as these two mole ratios. And at 32 to 1, we have particles and with a positive charged surface. So then we studied the production of antibody in the blood plasma. And we see from this chart, the mice were vaccinated with the polymer with a shorter chain and OVA nanoparticles, and produce the high amount of antibodies as compared to the polymer with longer chain and complex with the OVA. And from actually this chart, we can see that when we use poly-IC and together with the OVA, and there was a significantly high amount of antibody produced, and as compared to those actually stimulated with the polymer and OVA nanoparticles alone without the use of poly-IC. And another phenomenon we observed that in this column, and when you compare this column and to this column, and we see the antibody and production was quite similar, and for the polymer and aluminum as well. This is actually exciting for us because we hope actually to use the polymer as an alternative to aluminum as a vaccine adjuvant. So then we deliver the OVA and poly-IC in different nanoparticle formulations, and we found that at these two doses for poly-IC, the nanoparticle induced, the combination of nanoparticle induced a higher amount of antibodies as compared to the nanoparticles actually formed from the polymer and OVA without the use of poly-IC. And this is actually further proof that the use of poly-IC can further enhance antibody production in mice blood plasma. So from the biodistribution study, and we found that the biodistribution of OVA when delivered by the polymer is very similar to OVA alone without the use of the polymer. However, we did observe a high amount of OVA present in the injection site, which is also the site for inducing the immune response, and is actually much higher, and as compared to the OVA intensity in the skin area and induced by OVA alone at all time points from 0.45 hour and to 48 hours. So we isolated lymph nodes from the mice, especially from these two regions, and for inguinal lymph node and also mesenteric lymph node. In both regions, especially for lymph nodes, we see the both OVA and also the poly-IC were uniformly distributed in the lymph nodes. And we also observe the overlap of OVA and poly-IC. And in the region, we especially observe the yellow regions here. This is overlap of a red region and green region. As I mentioned previously, and these two formulations were injected at the same site, but actually at a different time point, which is actually 5 minutes apart. But we're still able to deliver both OVA and poly-IC to the same region of the lymph node, which is really exciting for us. So then we look at cellular uptake, especially by the dendritic cells and also macrophages, and for both OVA and also poly-IC. We observed that the uptake of OVA complex with the polymer was significantly higher compared to free OVA by dendritic cells, although we did not observe a significant difference in the uptake of OVA by macrophages and in our study. And for poly-IC, because it is an analog of double-stranded siRNA, and so it is very difficult for cells to take up without any carrier. So we see when we use our polymer as a carrier, we can significantly enhance cellular uptake for both dendritic cells and also macrophages. So this phenomenon was further proved by confocal microscopic study, and we see the greater intensity of a green region in dendritic cells by the polymer OVA nanoparticles as compared to OVA alone. And also we see the higher intensity for poly-IC in both dendritic cells and also macrophages and by the polymer and poly-IC nanoparticle formulation as compared to poly-IC alone. So from these actually findings, we can confirm that the use of the polymer can enhance cellular uptake by both dendritic cells and also macrophages. So then we studied long-term immunity against the cancer at the 9-month post-vaccination. And so the mice were challenged with the cancer cells at 9 months post-vaccination. At that time, the mice actually were already 13 months old, which indicate the middle age for these mice. And the tumor size of the mice that were vaccinated with these two formulations, including both poly-IC and OVA, and is much smaller, and like these two is much smaller as compared to these two formulations. Like the first one, this one is actually OVA alone, and this one is a combination of OVA and poly-IC. And on the other hand, we also see when we deliver both OVA and poly-IC in one particle formulation, we only see the smaller tumor size at 19 days, and as compared to these two formulations. However, after 19 days, and there's no difference between actually this formulation and these two formulations, and especially these two formulations, we did not use any polymer as an adjuvant. So, and more importantly, we also observed the tumors in the mice vaccinated with the polymer OVA nanoparticles, which is here, is actually were significantly smaller than those in the mice vaccinated with the OVA alone. So with this actually further confirm the polymer has actually better, has a very good actually, a very good efficacy in terms of inducing immunity against tumor challenge. So then we also studied the long-term immunity and in terms of antibody amount in the blood plasma at 9 months post-vaccination. And we see here the cationic polymer really play an important role in increasing the vaccination efficacy as all the formulations, starting from group 1 to group 3, and resulted in a significantly high amount of antibodies produced as compared to group 5 and group 6. And group 5 is actually injected by using the OVA and poly-IC mixture, and group 6 were injected with OVA alone. And the polymer OVA nanoparticles, and especially for group 4, and induce a high amount of antibodies than the OVA alone. And so this is further proof the nanoparticles can induce a high amount of antibody and then the OVA alone. So we also observe that you see here, and the activation of CTL, cytotoxic T lymph nodes toxicity, and at 1 month post-vaccination. The reason for us to study this because the activation of CTL activity is really crucial in killing cancer cells through T-cell mediated lysis of cancer cells. And the CTL activity of the groups that were vaccinated with the formulations from group 1 to group 4, which actually containing the polymer adjuvant, were quite similar compared to the one actually containing alumina-based adjuvant as positive control, which indicates that the polymer actually that we use can be a promising alternative to aluminum particles. See, our study, and especially another study, we use guanidinium functionalized polycarbonate as a vaccine adjuvant. And here we use actually the 2 different polymers, and with the length, like the repeated unit of 20. And one is with the C2 spacer, the another one with the C4 spacer. We found that both polymers can complex OVA and also poly-IC into very fine nanoparticles, and with a nano size distribution and positively charged surface. And we injected these 2 polymer nanoparticle formulation and into the mice at 2 different time points at day 0 and 4 weeks. And we took blood samples at 4 weeks and also 7 weeks post-vaccination. And we found that at 4 weeks formulations in the group 2, 3, and also 4 and 6 resulted in a significantly high amount of antibodies produced as compared to OVA aluminum and also OVA control group. And also we see from the group 3, the polymer with the C2 spacer led to a high amount of antibodies produced than aluminum particles at group 8. At the group 7, we observed that the only formulation in group 4 resulted in a significantly higher amount of antibody response as compared to OVA aluminum particles and OVA the control group as well. And however, we did not see any significant difference in the amount of antibodies produced between the group 3 and group 5 and the group 8. And this is quite interesting to us. And then we observed the tumor, the challenge experiment. And then the mice were actually challenged with the tumor cells at 7.5 weeks post-vaccination. And the formulations in the group 5 and also group 6 resulted in a significantly reduced tumor progression and compared to the OVA, the aluminum, and also the OVA control group. And so this is the OVA control group and group 7. And OVA aluminum particle group is group 8 is over here. So you see that the tumor size, there is a significant difference in the tumor size between these two groups and with this group. And so actually we can actually conclude that the group 5 and special group 5, we use the polymer with the C4 spacer and had actually much higher the immunization response as compared to aluminum, because we observed the stronger tumor rejection in the mice which were vaccinated by the polymer OVA nanocomplexes as compared to the mice which were injected by the OVA aluminum particles. So then we understand from our previous the experiment, actually the polymer did not induce a high amount of antibodies as compared to aluminum. But why we observed the beta tumor rejection in the mice which were vaccinated by the polymer OVA, the nanoparticles, and then we studied the cytotoxic T cell toxicity at 7 weeks post-vaccination. The cytotoxic T cells were retrieved from the spleens of vaccinated mice, and then we co-culture these cells together with the tumor cells and to check for cytotoxic activity. We observed that a significantly higher cytotoxic T cells activity from the cells treated with the groups 2, 5, and 6 formulations, which actually indicated here 2, 5, and 6 formulations as compared to the group 8, which were actually vaccinated using the aluminum OVA particles. So this could be the reason for the reduced tumor progression in the mice vaccinated with these 3 formulations, especially vaccinated with our formulations is like the polymer with the C4 spacer and the OVA nanocomplexes. So in summary, as a vaccine adjuvant, the polymer containing the block of cationic polycarbonate and another block of hydrophobic polylactide is actually quite comparable with aluminum particles. And then the polymer, especially guanidinium polymer with the C4 spacer, is more effective than aluminum in inducing immunity in mice. So this is a really exciting the findings for us. We will further actually study actually especially by using therapeutic vaccine and compare our polymer and aluminum as well. We also welcome collaboration with others who have actually new vaccines, especially against COVID-19 pandemic. Thank you very much for your attention. And I would like to end my presentation by giving thanks to, especially to Ashley, who did all the in vitro and in vivo study and with assistance from the Yanming and also Shujin, who is not here, and the Chuan, who is also not in the picture, and he did all the polymer synthesis. And I would also like to thank the other group members shown here and for their contributions to this study. And this work was funded by our institute, which is Institute of Bioengineering and Nanotechnology. It's under the arm of Biomedical Research Council, an agency for science, technology, and research, Singapore. And thank you for your attention. I look forward to seeing you at the conference. Thank you.
Hello, Professor Harashima. It's good to see you. And
Hi.
And so, hello everyone. I got several questions, 2 or 3 questions for each speakers. That I will ask a question instead of audience. And the first 3 questions are addressed to Professor Harashima. So I ask questions. First question is that the audience mentioned that he wondered how therapeutic agent, DNA or siRNA, can be selectively entrapped in manned nanoparticles.
Sorry, I couldn't catch your point.
Okay,
please ask me again.
Okay, the first questions, the audience said, I wonder how therapeutic agent, DNA or siRNA, can be selectively entrapped in MEND nanoparticle.
Okay. Usually, we are now thinking siRNA or messenger RNA as a therapeutic agent. So in case of these nucleic acid or messenger RNA, electrostatic interaction is the driving force of packaging into the MEND. So, encapsulation efficiency is very high, more than 90%. So very easily encapsulatable using microfluidic method.
Thank you. And they also wonder that what is the main difference between conventional cationic liposome and the MEND nanoparticles in terms of gene delivery?
The question is difference between conventional cationic system and pH-sensitive lipid. Okay.
Yes.
Yeah, pH-sensitive cationic lipid is a very wise system, originally created or proposed by Professor Peter Callis at British Columbia. This pH-sensitive system at neutral pH, the cationic lipid is neutral. Therefore, no need to introduce a lot of PEG into the system to shield the cationic charge. But once internalized into the endosome, acidic pH, then proton associated and the system becomes cationic. Then easy to fuse, interact with endosomal membrane and fuse, exert fusionic activity. So this the advantage of pH-sensitive cationic lipid is how to say very economical system by responding the circumstances, pH, especially pH.
Okay, thank you. And the last question is that they wonder how can be the synthetic cationic lipid metabolized? It might be different from the natural lipid. So that synthetic cationic lipid, how can be metabolized in the body?
Okay, that's a very important question from the viewpoint of side effect. Now we are making the 3rd generation, which is biodegradable. Therefore, toxicity is very little. However, the 1st generation or 2nd generation, they were not biodegradable. Therefore, they accumulated liver and spleen, and that could be a cause of toxicity. Therefore, when we design a pH-sensitive cationic lipid, we introduced biodegradable ester bond as a biodegradable component, we introduced ester bond into the pH-sensitive cationic lipid.
Thank you very much, Professor Harashima. Next question is questions addressed to Professor Rupenthal. First question is that for the first example, how specific is the sentry to the syndecon 4? And are there any barriers for sentry in binding with the syndecon 4? That is the first question.
That's a very good question. So we haven't done a lot of in-depth studies, but we've looked at, or a previous group that developed or investigated further on the cell-penetrating peptide, they specifically blocked syndecon 4, and then they didn't see any uptake. So it seems to be relatively specific. There's we couldn't see too much non-specific uptake, or they couldn't see too much non-specific uptake when they blocked the receptor with a specific blocker. But I would certainly like to do some more studies in terms of what the uptake kinetics are, and if there are any limitations to the binding in the future.
Thank you. And another question is that on the data which XG19 reduced the CMV lesion volume, was the complete recovery able to see?
Sorry, was the?
There is, in case of the XG19, they reduced the CMV lesion volumes. So do you see any complete recovery?
So not within the time frame that we investigated. Obviously, with the lesions, you do get recovery over time. This is, it's an acute injury model that we can see differences at a certain time point. If you do follow them over time, you will probably see faster recovery in the XG19 group compared to the non-treated group. But even in the non-injured animals, eventually, those lesions will be healed. Although on a molecular level, you might still see more scarring and more inflammatory markers upregulated in the non-treated animals. So it's in terms of the animal numbers, we just really looked at the time points where we were able to see a difference in terms of the action of the drug, rather than looking at the complete recovery, because we didn't really know when that complete recovery might be. But yes, in the future, you can certainly do more studies, especially with the OCT and the fundus imaging, where it's non-invasive imaging, where you could follow the animals towards the end.
And last question is that have you ever compared with Avastin or combination with Avastin?
No, we haven't done that yet. And we get that question quite a lot, comparing it to the gold standard. So again, that's something that we definitely have taken into account, and we're looking to introduce in a future study.
Okay, thank you very much. Next questions are addressed to Professor Ki-dong Park. First question is that it seems to need the enzyme such as HRP or H2O2 for in situ forming of hydrogel. For in vivo test or clinical situation, should we concern about HRP or H2O2?
Yeah, exactly. Around 10 years ago, we reported our hydrogel system. We got this kind of question so many times. So at that time, we didn't have enough data. So anyway, the answer is definitely it depends on a concentration. And I want to answer, there are our product are waiting for a clinical application in a KFDA. We have enough safety data. So we learned that hydrogen peroxide toxic, HRP can be a human applicable. Definitely, it depends on a concentration.
Thanks. And the next question is that when we design the hydrogel for encapsulating the cells in hydrogel, which factor should we consider compared to deliver chemical drug or peptide?
Yeah, that's a good question too. When we deliver cell, most of research scientists are struggling how to maintain a cell viability. That's the most important issue. So cell viability depends on so many different parameters. So many matrices, many scaffold are reported to be cell comparable. And how long can we maintain cell viability, and how can we maintain a cell in a wound site? That's most important question. So hydrogel should be a cell comparable, definitely a safety issue, and this one should stay in a defect site for longer time. That's why stem cell therapeutics doesn't show a high efficiency.
Okay, thanks. And the last question is that hydrogen peroxide responsive hydrogel has been showed astonishing result to diverse pathogenic status. Do they also construct cross-linking through the endogenous ROS reactions?
Yeah, this is also a good question. We had the same question. So when we spray our hydrogel on skin ulcer, etc., can we get a hydrogel formation? Yes, it's possible, but it takes a longer time than we expected. So we have to add a small amount of hydrogen peroxide together.
Thank you very much. And the last three questions are addressed to Professor Yang. The first question is that macrophages and dendritic cells are major APC in our immune system. However, it is well known that dendritic cells are more important in antigen presentation and adaptive immune system activation. In this study, bone marrow-derived macrophages showed a higher cellular uptake of nanovaccine compared to bone marrow-derived dendritic cell. Do you think bone marrow-derived macrophages also have an important role in your formulation?
Hi, it's really so good to see you and Professor Byung and also Professor Park. And this is a very good question. And so I think it's actually quite important. It's a dendritic cells. Dendritic cells is the most important, but macrophages also, we believe, also play a certain role. And but we believe is dendritic cells play a key role in this vaccination. Yeah. But anyway, from the data is actually for dendritic cells, and the cellular uptake is also quite high. It's close to 70%. So not so bad.
Thanks. Second question is that what is the mechanism of cationic polycarbonate polymer inducing adjuvant effect? For example, due to agonism of TLR or STING receptor, or due to the enhanced accumulation of delivered antigen to lymph node. So that what is the main mechanisms of cationic polycarbonate polymer inducing adjuvant effect?
Yeah, it's very good question. Very good question, because it's a polymer conformed nanoparticle, and especially for poly-IC, and it's a very strong enhancement for immunity. And then for the OV8-wise also, it also can form particles by using the polymer. So I believe it's because of formulation of the particles so that we can deliver to cells easily, especially for cellular uptake. But let's see, we have seen the enhanced cellular uptake by dendritic cells and also macrophage. And on the other hand, we also observe the accumulation of the particle formulation in the lymph node. So that could be the reason it can deliver to the lymph node, especially for the polycarbonate with gonadinium function group. And you could see also very strong activity in terms of CTL cytotoxicity. So believe it can deliver to the lymph node and to activate the CTL cytotoxicity.
Thank you. And the Last question is that OVA antigen is complexed with cationic polymer by a charge interaction. Is it possible to apply this cationic polymer to deliver a small peptide antigen or a non-negatively charged antigen?
So this is really another very good question, and this we could for these polymers is really is good to deliver a negatively charged the antigens, and especially RNA, and for if we use RNA for vaccine mRNA, like this case for COVID-19, and so for other antigens like small peptide, which is not anionic charged, or even for other molecules, and we can design certain different carriers. I think most of the friends here, we are able to deliver, to design different carriers for specifically for certain molecules, like different type of peptide.
And thank you very much. And
thank you.
Everyone, thank you very much for your nice presentation and discussions. And take care of yourself in COVID-19 situation. Thank you. And now you are close to this first session. Thank you very much.
Bye-bye.
Thank you.
Thank you very much. Thank you.
Welcome to the second session of the 43rd KAST International Symposium. My name is Yu Kyung-ho from Seoul National University. I'll do the session chair.
Therefore, you have to read.
Pardon me? Okay, so like in the first session, we will collect the questions and online, and after all the session is over, we will discuss all questions in live session together. So let me introduce the speakers for session 2. So the first speaker will be Professor Bruno Sarmento from University of Porto. Professor Sarmento is the chair-elect of CRS International Chapters, and Professor Sarmento is an editor of European Journal of Pharmaceutical Science. And he will discuss about receptor-mediated delivery of multifunctional nanoparticles. Second speaker will be Professor Young-Bin Choi. He is from Seoul National University, and Professor Choi is a member of Young KAST. He will deliver implantable device story. The third speaker will be Professor Randy Milsny from Bath University, UK. Professor Randy Milsny is the prospective editor of Journal of Controlled Release, and he is also CRS Controlled Release Society College of Fellow. Professor Milsny will discuss how we can open tight junction. The last speaker, fourth speaker, will be Professor Dan Pierre from Israel. Professor Pierre is an associate editor of Journal of Controlled Release, and he had been the board member of Israel Young Academy of Science. So we will now start the first presentation of Professor Sarmento. Thank you.
Hi, good afternoon to all. First of all, I'd like to thank the kind invitation to be here today, especially to Professor Yu Kyung. This is actually a great honor to attend this meeting and to participate on this exciting event from the Korean Society. So my talk today is related with the oral delivery of anti-diabetic peptides, and I'm going to talk a bit about our work that we have been developing in our lab that involves the development and the validation of the receptor-mediated transport of functionalized nanomedicines for improving the intestinal absorption of anti-diabetic peptides. And why diabetes, and why do we have this interest in diabetes? So we believe that diabetes is still a huge health issue all around the world. So we know that roughly 10% of the world population is suffering from diabetes. And not only in terms of world population, but also in terms of expenses that the healthcare systems still have on control the symptomatology and control the effects of diabetes. So there is a huge potential in terms of developing of new therapies, develop of more efficient therapies, and also in terms of markets that we can target with new products. And just to come a little bit on the physiopathology of the disease. So we know now very well that diabetes is mostly related with the insufficient production of insulin by our body, or by the insufficient response to the insulin production that our body is able to release to the bloodstream. And depending on the severity of the diseases, we have type 1 and type 2 diabetes. And in type 1 diabetes, that accounts for close to 10% of the total number of diabetic cases all around the world. We know now that there is an autoimmune destruction of the insulin production beta cells on the pancreas, and that's why our body is not able to produce or to respond to the increase of glycemia with more production of the insulin. And that's why insulin administration, so exogenous insulin administration, is the most convenient treatment for type 1 diabetes. There's other alternatives that are being developed in the last few years, mostly in terms of clinical trials, namely the pancreatic transplantation or stem cell-based therapy that I personally believe will have a huge impact as soon as they are disseminated in the clinics. But still, there are some drawbacks associated with these cell-based therapies. And when we look at type 2 diabetes, we know it's a less severe condition of the disease, and we have different therapeutic approaches to control type 2 diabetes, but it is more and more used, and with better outcomes for the patients, the inclusion of GLP-1 analogs to control this disease. And these GLP-1 analogs are mostly peptidic-based molecules that still have some issues in terms of bioavailability, as we'll show you later on. So overall, we have a huge market for therapeutical peptides. The overall market for therapeutical peptides is very interesting in terms of the clinical outcomes all around the world. We have a very interesting percentage of number of peptides that are available for clinicians to treat different diseases, and the approval rate for therapeutical peptides, it's quite higher compared to the overall new chemical entities. And so I believe that numbers close to 10% are quite interesting. So we have a huge potentiality in terms of explore these kind of molecules. So if we have therapeutical peptides to treat diabetes, so why do we need non-invasive delivery systems? Most of these drug products are administered to patients using invasive routes, subcutaneous mainly. And this parenteral route, of course, is not absent of side effects. So we know that patients usually don't like parenteral delivery because it's painful. It's a chronic treatment, so it's psychological things that will have a treatment for the life. And also there's a risk of infection, and there's also a high cost of associated with the administration of these drug products. So in our perspective, the non-invasive delivery of antidiabetic peptides would facilitate the administration of the drugs, make patients more proud to adhere to the medicines, and also we believe there's still plenty of room for market competition if we have alternative routes of administration for well demonstrated antidiabetic peptides. But of course, peptides are drugs that are not easy to be administered with non-invasive routes. We know that they have intrinsically low permeability because of the ice molecular weight that they have, also due to the hydrophilic nature of the peptides. Also, they suffer from proteolytic degradation when we explore mucosal delivery. And at the non-invasive routes and focus on mucosal delivery, we have important barriers against the absorption of these proteins or peptides, namely the mucus and the epithelium itself. And when you look at the gastrointestinal tract, so we can easily identify the main barriers in terms of the stomach hard dish regarding the acidic pH and also the enzymatic degradation, and also at the absorption window of the intestine, where we clearly see the mucus, the intestinal epithelium, and the enzymatic degradation as the main barriers against the high absorption of antipeptidic drugs. Still, this is nothing that can be discarded. The fact that we have clinical trials in late stage of study, and also some products on the market that explore technologies, drug delivery technologies for oral administration of insulin or GLP-1 analogs. I draw your attention to Rybelsus, that is a tablet-based drug product that was approved a few years ago for the oral administration of GLP-1 analog. So it means that it's possible to have technologies to explore as much as you can specific features, specific properties of excipients of drug delivery products to provide significant amounts of bioavailability for these molecules. And that's what we try to do in our lab, explore nanomedicines to provide physical and chemical stability against the degradation of these peptides, to provide mucopenetration by modifying the surface of these nanomedicines. And also, at the end of the day, what we want is to have an enhanced intracellular delivery through the enterocytes in order that nanoparticles release their payloads or part of the payloads on the bloodstream. And we have a deep knowledge on how to make nanomedicines mucopenetrated, mucodiffusive, and also we have high experience in terms of produce systems that can have stability for the protein, and also they can target specific areas of our intestine. So the overall aim of the work that we developed in our lab that is related with intestinal delivery of antidiabetic peptides is on the availability of provide matrix drug carriers with suitable properties to assure the stability of the proteins that can somehow control the release of these proteins and also to modulate the mucodiffusion of the proteins to the epithelium. But of course, the main important aspect of our system is to provide pharmacologically relevant oral bioavailability for these antidiabetic peptides. So, a few years ago, we established a methodology that explores polymeric matrix, namely PLGA matrix. That is a well-established material that is FDA approval. So we know there are lots of drug products on the market comprise PLGA. So we want to keep as simple and as, from the regulatory point of view, as fast an approval as possible. So by using biomaterials or excipients that have acceptability from our perspective is an advantage. So we established methodologies to encapsulate insulin and other antidiabetic peptides that could be able to provide physical stability for the protein after encapsulation, and we demonstrated that is possible. So proteins like insulin retain the secondary and tertiary structure after encapsulated into PLGA matrix and after releasing from the PLGA matrix. And we also optimize protocols for the encapsulation and for the freeze drying of these nanomedicines in order to attain as much as possible the spectral similarity of the insulin, of the native insulin with the insulin that is encapsulated and released. So from here, we gain substantial experience on established methodologies that are safe and potentially provide bioactivity for the diabetic peptides. So from that, we explore different areas. And one of the areas that we are exploring is the transcytosis of nanoparticles containing insulin that could explore specific receptors of the intestine to have a perfect permeation to the full mucosa, to the mucus, to the epithelium, and reach the bloodstream. And one of the receptors that we are exploring is the FcRn. So we know that FcRn is the Fc receptor is a pH-dependent receptor able to or responsible for the transcytosis of albumin and immunoglobulins in general, not only in the intestine, but in other organs of our body, mainly at the level of endothelial cells. But at the intestinal side, the FcRn has this very nice factor that is the pH dependence of the transcytosis. So at pH close to 6, albumin or other ligands bind perfectly to the FcRn, and FcRn is internalized by cells, and then at physiological pH of 7.4, there's a release to the bloodstream of the ligands that were previously linked to the FcRn. So the approach is to have nanoparticles that somehow could be internalized to the enterocytes in a pH-dependent manner, and then being transcytosed to the basolateral site. So this was not new. We and others have been explored the use of the FcRn receptor with the exploitation of different ligands like fractions of immunoglobulins or the albumin itself. So we have this in mind. But the fact is that Fc fragments or human albumin may have limitations in terms of the binding capacity to the FcRn. So we have a very clever gate to enter the bloodstream to the intestine, but probably we are not exploring as much as we can this gate. But luckily, a few years ago, we established a collaboration with Jan Andersen at Oslo University Hospital in order to explore the capacity of some albumin variants that they produce. So what they did, they slightly modified the structure of the human albumin in order to have variants with higher affinity to the FcRn and lower affinity to the FcRn. So in that sense, they did some biotechnology approach to have this high affinity variant and low affinity variant, and they demonstrate that this modification of the human albumin may have different binding capacities to the FcRn, but as albumin variants alone, not in the context of functionalization of nanoparticles. So we took the idea of explore PLGA-based matrix to encapsulate insulin, provide mucodiffusivity of these particles by pegylating the surface of PLGA nanoparticles. We know that the pegylation is probably the most universal strategy to provide mucodiffusion to nanoparticles. We have other strategies, but in this case, we are pegylating the particles. And on the top of the PEG moiety, we are anchoring albumin or albumin variants in a site-oriented manner in order to retain the binding capacity of the albumin to the Fc receptor. So this is the overall approach that we are trying to explore. So very easily, we set up nanoparticles, we characterize these nanoparticles. So just to explain it in more detail. So we encapsulate insulin into PLG nanoparticles, and we functionalize these nanoparticles with wild-type albumin with this variant that is the KAHK, and also the KP variant. So just make longer story short, Ka is a low binding variant and KP is a high binding variant. So the idea is to have a comparison between these variants with the wild-type albumin, and of course, systems that are not functionalized at all. So we are talking about particles roughly around 150 nanometers in size, particles that are almost negatively neutral in charge, slightly negative, but this is mostly because of the overall negative charge of the albumin on the surface. Particles that can encapsulate close to 80% of the insulin that we are using and can bind more than 90% of the albumin that we are trying to attach on the surface of the nanoparticles, resulting in drug loadings close to 10%, which is quite acceptable for a very potent drug as insulin. So then what we did, we confirmed that after functionalized nanoparticles with different variants of albumin, we didn't compromise the binding capacity to the FcRn. So this is an in vitro assay by a sandwich ELISA. And what we compare was the binding capacity of the albumin variants alone and after attached to the surface of nanoparticles. So basically we are not losing binding capacity, which is at this stage was a very good result. Then we did some physical chemical characterization. So we have particles that release very low amounts of insulin with the time, as we've seen here. Particles that, again, are able to retain the structure of the insulin inside the PLG matrix with a very high level of spectral similarity between native insulin and insulin that is encapsulated. So as we confirmed that the binding to FcRn was not compromised, then we performed a permeability test using a monolayer based model. In that case, we use a cell type that was modifying to express human FcRn. So we overexpress the human FcRn of this and these AK lines, the cells, and then we quantify the transport of albumin to the monolayer. And again, we demonstrate that the albumin alone and after functionalizing on the surface of nanoparticles, retain their binding capacity, and in that case, their transcytosis capacity. So we have a very nice pattern of increasing the KP variant, increasing the transcytosis of nanoparticles compared, for instance, with wild-type albumin or with nanoparticles that are not functionalized at all. Okay, in the meanwhile, we have this very interesting experience that was in collaboration with European Space Agency to test whether or not the transcytosis capacity of these nanoparticles were dependent on the gravity conditions that human body may be suffering. Of course, this is an in vitro test, but we can interfere or can correlate then with in vivo. So this was an experiment that we performed. So we look at the permeability of the albumin in different hypergravity conditions from 1 to 7.5 Gs. And interesting, what we observed was that permeability of the nanoparticles, or at least of these functionalized nanoparticles, were not dependent on the gravity. So this is a very side study that we are still exploring, but we don't know any previous result demonstrates the absence of effect of hypergravity on the permeability of nanoparticles. So that's quite nice. Then we perform ex vivo permeability of these nanoparticles using pig tissue, pig intestine. So in collaboration with Giovanni Traversco in MIT. So what we did, we select specific parts, segments of pig intestine, and then we quantify the relative transport of this human albumin functionalized nanoparticles. We compare again the free albumins and the nanoparticles functionalized with the same albumins, and again we see a trend in terms of increasing the binding capacity of the albumin variants and the transcytosis capacity of these nanoparticles. So all of this bunch of data gave us some motivation to end up in an in vivo study, but we have an issue for the in vivo studies that we were modifying these particles with human albumin. And using animal models, we may have some influence of the cross-reactivity or the binding effect of the different interspecies difference. So luckily we found this mouse model that was modifying to express human FcRn instead of mouse FcRn. So this mouse model allows us to test the efficacy or the effect of the functionalization of nanoparticles with human albumin. So we modify this animal model, so we make it diabetic with the STZ protocol. And after oral administration of the particles, we quantify the levels of glycemia with the time, so upon the oral administration. So if you look at the glycemia data, and of course compare with subcutaneous control of insulin, we see an interesting difference when we formulate or when we administer nanoparticles functionalized with our KP variant of the albumin compared to the wild type albumin. Looking at pharmacokinetic data, we have close to 6% of pharmacological bioavailability of insulin. But what is more interesting here is that we have close to 20% of hypoglycemic response when we administer these nanoparticles containing insulin compared to the insulin subcutaneous. So there's no doubt that the modification of these nanoparticles provide the mucosal diffusion capacities and targeting ability to FcRn result in an enhanced effect of the oral absorption of the insulin. Well, this is true for the insulin, but we may consider other antidiabetic peptides. And as I mentioned before, we have this proof of concept now on the market, the Ribelsus, that is a tablet that comprises semaglutide, so a GLP-1 analog. And this modification of the molecule provides a very interesting value for oral bioavailability, but still is not perfect. We are dealing with a drug product that results in close to 1% of oral bioavailability. So what we think is that if we could adapt our previous strategy to target nanoparticles to FcRn, we may have a similar effect in terms of improving the bioavailability of the semaglutide. So the strategy here is to have a PLGA-based nanoparticle that is modified on the surface with PEG to provide mucodiffusivity to the particle and also functionalized to the FcRn. But in that case, we are not using human variants or human albumin variants, but we are exploring peptides as ligands to the FcRn. And why? Because peptides are shorter in size, they probably will be easier to chemically anchor on the surface of nanoparticles. And we have some preliminary data that show us that specific sequence of peptides are more efficient in terms of binding the FcRn compared to albumin or albumin variants. So we are still starting this project, but we are very confident that we'll have very nice results quite soon. And why we believe so? Because previously we explored not semaglutide, but GLP-1 itself in terms of oral bioavailability and whether or not the oral administration of GLP-1 could result in significant values of bioavailability. So in this work, we encapsulate GLP-1 in PLGA nanoparticles. We modify the surface of the nanoparticles with CPP, so we use CPPs to target the enterocytes. And we encapsulate these nanoparticles into pH-dependent microparticles. So the idea here was to have a system that could cross the stomach without releasing nanoparticles, so without compromising the stability of GLP-1, and also a system that could at the same time release a second drug, that is this DPP-4 inhibitor, in order to avoid the premature degradation of the GLP-1. We know that GLP-1 is degraded by DPP-4, so if we can somehow inhibit the action of DPP-4, we can increase the half-life of the GLP-1 upon administration. So this was performed in a microfluidics device. So we have microparticles that were pH dependent in terms of degradation, in terms of release, always the same trend. So we have GLP-1 release that was absent at gastric pH, but was controlled and in a sustained manner at intestinal pH. But the DPP-4 inhibitor was immediately released even at gastric pH, which is positive in our case because we want to have a previous action of inhibitor and only after action on the GLP-1. And before we go to in vivo studies, we just briefly test whether or not these modified nanoparticles on the surface could interact with enterocytes. So here we can see that when we have the PLGA nanoparticles modified with the CPP, the level of interaction with intestinal cells is much higher compared to the PLGA nanoparticles that are not modified. And we can also quantify this fold increase by facts. So there's no doubt that the CPP modification of the surface of the particles increased the interaction of nanoparticles with enterocytes. And when we go to an in vivo proof of concept, so in that case, we use a type 2 diabetic animal model because the GLP-1 only acts on type 2 diabetics, not in type 1 diabetic. It was very interesting to observe that we have a hypoglycemic effect, not immediately, but after roughly 2 hours of administration of these nanoparticles orally. So we have a significant decrease on the levels of glycemia for the formulation that combines the GLP-1 and the DPP-4 inhibitor. But we didn't observe the same trend for the formulation with just GLP-1 or the formulation with just the DPP-4 inhibitor. So meaning that the synergic effect was crucial to have this decrease of the glycemia. And the pharmacodynamic data were overlapped with the levels of insulin that we observe in the blood of these mouse. So after 4, but mainly after 6 hours, we are able to observe insulin that was released. So meaning that the GLP-1 was able to reach the bloodstream to attain the beta cells on the pancreas of these type 2 diabetic animals and provide extra secretion of insulin that afterwards result in the decrease of glycemia. So we have a very nice set of data that demonstrate how the GLP-1 was absorbed and how GLP-1 was responsible for the secretion of extra levels of insulin in this case. Just before finish, let me talk about another project that we are running at the moment in the lab that is related also with anti-diabetic peptides, but not the conventional peptide. We are exploring here the action of the cathelicidin LL37, that is a well-known antimicrobial peptide. It's a peptide with demonstration in terms of demonstration in terms of action to leukocytes and epithelial cells. And there's also some evidence that this cathelicidin LL37 may act on the beta cell and sensitize these beta cells to secrete insulin. So this was not new, but what is new is that we have a collaboration with another group here in Portugal that developed this derivative, that is the LLKKK18 peptide. So we have here the sequence. Basically, the LL18 is a fraction of the original LL37. It's a smaller peptide, but we believe that it's able to retain the same trends in terms of biological actions of the peptide. So for the LL37, the action in promote beta cell function was already demonstrated, but not for this smaller fragment, and that's what you want to test. And again, we are trying to encapsulate, or we are encapsulating the LL18 in PLJ particles. We are pegylating these particles, and we are targeting these nanoparticles to the beta cell. So how can we target these particles to beta cells? So our strategy here is quite simple. So we are functionalized the particles with exenatide. So exenatide is a GLP-1 analog, is used in the clinics to treat type 2 diabetes, but our aim here is not to use the exenatide as a drug, is to use exenatide as a ligand. So we are modifying this exenatide with a terminal cysteine to have the site-oriented conjugation of the particle, and then produce the particle, encapsulate the drug, and see whether or not this drug is taking action on the beta cell sensitization. So this is roughly what you want. So just very preliminary data on this project. So we have particles that are quite small, roughly 100 nanometers in size. We have very nice values for association efficiency between 70 to 80%. This is quite positive, even considering that we have a very hydrophilic peptide, but this methodology that we are adapting for the double emulsion is able to encapsulate high levels of the exenatide, of the LL37, and also we are able to functionalize the exenatides on the surface. This means that the particle that you have there on the process of the functionalization, we are not releasing the encapsulated drug. So these particles are round in shape. So we just did a very fast physical chemical analysis to compare the morphology. We know that the peptide is released in a controlled manner. So we have release profiles that reach 50% of the drug up to 150 hours. So it's quite good in terms of providing long-term action for the LL18. And what we very recently demonstrated was that when LL18 is encapsulated in the particles, in the functionalized particles, we see in certain conditions the release of insulin from beta cells. So this release is dose dependent, of course, and also is glucose dependent. So with higher concentration of glucose, we have a higher release of insulin. These data were very, very encouraged. We are very happy with these results. These are in vitro results yet. So we are now trying to establish a protocol to demonstrate this in vivo. But with this experiment, we demonstrate not only the biofunctionality of the peptide, the LL18 peptide, but also the capacity of the exenatide to act as a ligand to target nanoparticles to the beta cells, which is a very nice achievement. So just to sum up, I hope that I have demonstrated to you the potentiality of nanoparticles, specifically PLJ-based nanoparticles, in terms of provide favorable conditions for encapsulate anti-diabetic peptides. We have proof of concepts using insulin and GLP-1 analogs, but potentially many other peptides. And in particular, we explore the FcRn transcytosis capacity that is present at intestine to provide systemic absorption of insulin, and we expect also for GLP-1, and also to modulate the diabetic conditions. And we also have we also demonstrate how nano-microsystems may act as very promising technological platforms to provide dual control release of GLP-1 and GLP-1 analogs, and also DPP-4 inhibitors. That this is a very clever and very elegant work that we demonstrate. And we hope to have very soon evidence that anti-diabetic peptides as catalytic derivatives could also be explored in the diabetic treatment. So just to finish, I want to acknowledge all the members of the group, the present members and the former members of the group that help us in terms of raising this exciting data. Also our collaborators all around the world. I would like to highlight in here Jan Anderson from also Oslo University Hospital, also Elder Sandstorm University of Helsinki, and Giovanni Traverso from MIT. And of course, I thank all your attention. Again, thank you very much for your invitation, and I'm very happy to address any questions that you may have.
Okay, hello everyone. I'm very glad to have this opportunity to talk about my work here today. I'm Young Choi. I work at the Seoul National University College of Medicine. I guess I'm the only device guy here today. Today what I'm going to talk about is a batteryless implantable devices for on-demand and pulsatile drug delivery. The page doesn't seem to. Okay, so today what I would like to talk about is the batteryless implantable devices enabled with on-demand and pulsatile drug delivery. And what we were interested in our lab is about self-injectable drugs. And then they are mostly like large molecules such as insulin and growth hormone. And they need to be delivered in a pulsatile manner to mimic physiological secretion profiles. However, it's not convenient for the patients because they need multiple daily injections. The injections involved with needles, that's why it's painful. And sometimes it's very difficult to fit the dose all the time. So it becomes sometimes erroneous. And to resolve this issue, and for example, for the insulin delivery, a continuous subcutaneous infusion device has been already developed and in use in clinical settings. And however, the patient still needs the needles to be attached in the skin for a long time. And the device is slightly bulky and it's external, so the device needs to be carried all the time with the patients. And another issue to be resolved is, so people suggest, is implantable devices. So after one-time implantation, the device can work like a mechanical machine to realize a pulsatile or a continuous drug infusion. And so the patients can even control the device, implanted device itself, so that the patient can still achieve an on-demand or patient-driven drug infusion. Actually, to meet this need, the device always contains the battery or electronics inside, so that those are together to be implanted in the patients. So it becomes slightly bulky for implantation, and then when the battery lifetime is out, the patients always need explantation surgery. So it becomes a major issue again. So we have this, given those, we have this design criteria. So what if we can actuate the device without a battery? However, we still need to achieve on-demand and pulsatile drug delivery. So we need a non-invasive means. Without skin punctures, we still need to deliver drugs in a pulsatile manner. And also, if for the chronic diseases, then the device needs to be used semi-permanently. So these are the suggestions that we had. So what if the device can be driven by a magnetic force? As you may already know, a magnetic force can penetrate into the body non-invasively. What if we can make the device to release the drug at the time of near-infrared light irradiation? As you again may know, the NIL light can penetrate into the body without invasiveness. And for the chronic disease treatment, after implantation, the pump or the device needs to be refilled with a simple method, so the device may need to be equipped with a refill port. So today I'd like to introduce two devices developed in our lab. The one on the left is for growth hormone deficiency therapy, and the other one on the right is for the treatment of diabetes mellitus. So growth hormone deficiency, it's a medical condition caused by not enough growth hormone. So the therapy itself is more like a supplementary therapy. So the growth hormone itself is delivered from the outside of the body, again, which needs multiple daily injections. It involves with needles. And for growth hormone deficiency treatment, the patients are mostly, many of the patients are children, so the parents still need to carry those needle injections all the time. So it's still painful and it's not quite convenient. So our idea was, what if we develop a device that can deliver the drug only at the time of non-invasive means applied from the outside of body? So this is the device we suggest, which contains a lot of drug reservoirs, each containing a same amount of growth hormone inside. And then on top of that, we would like to cover them, perfectly seal them, with a stimulus responsive membrane. So this membrane, it can be designed to be ruptured only when the NIL, near infrared light, is irradiated. And because there are many reservoirs in the implanted body, and we have to align the NIL light only at the specific reservoir, so we have to have some means to locate and align those NIL irradiation from the outside of body. So that's why we have those two magnets embedded in the device with the two different polarity. So on the right side, you can see the external guide, which can contain the two magnets again with a opposite polarity, so that this can be attached from the outside of the skin. And then the external guide can contain a lot of holes to be aligned to each of the reservoir, so that we can apply the NIL light at a specific location. So the most important technology here is a stimulus responsive membrane. We used the composite of a POSS polymer and reduced graphene oxide to prepare this membrane. And so the polymer itself here has got a characteristic that it can be torn when the temperature rises. And however, NIL light cannot directly increase the temperature. That's why we had the reduced graphene oxides, RGO nanoparticles inside of this membrane. So when this composite is formed and the NIL light is applied from the outside of the skin to reach the membrane, and then the light will excite these RGO nanoparticles, which will generate the heat, and the temperature rises, and then this will rupture the polymer. So these are the several different conditions that we prepared for this stimulus responsive membrane. On the left side, what you can see is the membrane made of the polymer only, without the reduced graphene oxide nanoparticles. And then after NIL light irradiation, it is intact. It wasn't torn. And however, with some amount of RGO nanoparticles applied, together embedded inside of the membrane, as you could see, there was a partial opening. However, if it's not homogeneously distributed, if the nanoparticles are not homogeneously distributed, we could not fully open this membrane. However, on the very right side, you could see that the membrane is quite well composed of the reduced graphene oxide nanoparticles, showing a slightly black color. And then when we applied NIL light, the reservoir could be fully opened. So this is the optical microscopy images of our device on the left side. As you could see, there are like different membranes, different reservoirs, each containing the same amount of growth hormone. And then it is fully covered with a stimulus responsive membrane. And then on the right side, there's an external guide that we designed for now as a prototype. And as you could see, there are several holes available there, so that each of the hole can be used to apply the NIL light to be aligned to each of the specific reservoir that we would like to apply on. So this movie can show some insight about our device. And so this is the device that we made with a membrane covering the whole reservoir, and when we implant it. subcutaneously. And then afterwards, as I mentioned earlier, there are magnets inside so that the external device can be well aligned and attached on top of the skin. And then for each of the reservoir, we can apply NIL light irradiation. So we run it quickly so that we can try to open all the reservoirs made in the device. And then, so after we apply all of the NIL lights to all of the reservoirs, and we took out our device, as you could see, all of the reservoirs could be fully opened. So we first tested our device under in vitro environments where we fully immersed the device inside of the pH 7.4 PBS, that's 37 degrees Celsius. And as you could see, when there's no irradiation, there was no leak of growth hormone, meaning that we could fully and quite well sealed the reservoir with a stimulus responsive membrane. And only at the times of irradiation, as you could see, the exact, quite reproducible amount of growth hormone could be released to the release media. And to test the feasibility of our device, we employed hypophyseectomized rats. It's an animal model with a growth hormone deficiency. And we had 3 different groups. The control group are the rats with no treatment. And the other group is with the subcutaneous injection groups, where the rats were subcutaneously injected with the same dose of growth hormone every day for 14 days. And for the device group, are the rats implanted with our device. And in this case, we did not do any needled injections, rather than just applied the NIL light non-invasively from the skin using the external device. And the dose delivered were the same with the subcutaneous injection and device groups. So this is the short time in vivo profiles. And on the left side, you could see the plasma growth hormone concentration profiles. And for the subcutaneous injection groups, as expected, after injection, the concentration goes up very high and it drops down quickly as during the about 300 minutes. And then what we need to observe here is that with the device group, only with the non-invasive NIL irradiation, as you could see, this short-time pharmacokinetic profile is quite similar to that of the subcutaneous injection group. And then as a pharmacodynamic profile, we measure the plasma IGF-1 concentration. And as you could see, those 2 profiles between the groups of subcutaneous injection and device, they were similar. So again, probably our device could do the same drug efficacy applied to the body as done with the conventional subcutaneous injection. And we also performed the long time in vivo pharmacokinetic and dynamic profiles. And as you could see, we only measure the maximum concentration here. And for 14 days of experiments, and when you measure the blood concentration at scheduled times, those maximum concentration observed for the 14 days were similar between subcutaneous injection and device groups. And again, the plasma IGF-1 concentration were again similar between those 2 groups. And as this model has got a growth hormone deficiency, as you could see from the control group, this does not grow. The animals do not grow. So the body weight gain was almost the baseline level during the whole testing periods. However, when this growth hormone was delivered every day using both subcutaneous injection and device groups, as you could see, those growth pattern, body weight gain patterns were quite similar between 2 groups. And then when we observed the histopathologic tissue evaluation results from this picture, we obtained the tissues around the implanted device and then did some staining, and then we got the evaluation from a pathologist in our hospital. And then they said that an overall inflammatory and foreign body reactions were minimal for all tested tissue locations around the device, meaning that our device could be used safely after implantation. So for the conclusion of the first, we have proposed an implantable multi-reservoir device with a stimulus responsive membrane for on-demand and pulsatile delivery of growth hormone. More importantly, we did not use needled injections. Rather, we used non-invasive NIL light irradiation to perform the similar pharmacokinetic, pharmacodynamic profiles as done with the conventional injections. Therefore, we thought that this device herein could be a promising and minimally invasive strategy and as an alternative for on-demand and pulsatile delivery of growth hormone. And for the second device, we focused on the diabetes mellitus, and this was well introduced by the previous speaker. And so as you could see, the statistics was projected to be about 642 million people will have diabetes. And so, well, we have big needs, and also we can think of this needs in market too. And again, this is done, this insulin therapy is also done with multiple injections, and it involves with needles and pains. Therefore, we would like to make the pump that could infuse the insulin only at the time that we need. So we prepare this type of device, which could be actuated by a magnetic force. As you can see, we have a plunger and barrel, and plunger is a movable part. And then at the stationary face, plunger and barrel contains the magnet. The black parts show the magnets. They have the opposite polarity, so that's why they were stuck together, so they don't move. However, when externally from the outside of skin, if we apply the stronger magnet, and then the plunger was pulled, and then the insulin solution contained the drug reservoir will be sucked into the barrel, and then when we remove the magnet from the outside of skin, again, the plunger was pulled down to the magnet in the barrel, pushes the drug solution to make that infused outside of the pump. So this slide will show you some insight of our device. And for now, for the visibility, the pump is filled with an ink. And the simple structure works like this. As you can see, external magnet applies, plunger moves upward, and then the drug solution in the reservoir is going into the barrel. And then when the external magnet is removed, and then plunger moves downward and pushes the drug solution through the check valve. And as you could see, this is more like a water gun with which can infuse an exact volume of liquid all the time. Well, only at the times when the magnet is applied. So as you could see, the uniform droplets could be formed. So if we feel different concentration of insulin solution inside of the pump, then we could also modulate the dose for each actuation. So again, we test this pump, and while this was fully immersed in the PBS at pH 7.4, and when you applied multiple actuations, and then you could see the reproducibility of the dose of insulin released at the release media. And then when we immerse the pump for a long time, for like 30 days, and when there's no actuation, when there's no magnetic field applied from the outside, and then there was no leak of insulin. However, only at the times of actuation with magnet application, as you could see, there's a reproducible insulin release. And to test this feasibility, at this time, we employed STZ-induced diabetic rats. This is basically a type 1 diabetic animal model. So this animal doesn't have any ability to produce insulin properly. And at this time, we have 4 different groups. The control groups are the diabetic rats with no treatment. And the subcutaneous injection groups are the diabetic rats treated conventionally, by a needle syringe injections of insulins. And MDP1A group is the rats implanted with the device, and then the device was actuated by a non-invasive magnetic field force from the outside of body. And MDP1A2A group is the device that was actuated for 11 days, but afterwards we increased this consecutive actuations to 2 to show the dose controllability with simply by varying the actuation numbers. And this is the long-time in vivo insulin concentration profiles. And as you could see, for the subcutaneous injection groups, after we apply, we administer insulin of the dose, and then we measure the maximum concentrations. The profiles were quite similar for the whole testing periods. And then for the MDP1A group, as you could see, before 11 days, those maximum concentration profiles were quite similar to that of subcutaneous injection groups. However, interestingly, after 11 days, these plasma concentration dropped. We thought that because when we implanted this bulky implants pump, then there would be a fibrotic capsule formation around it, which hinders the drug released from the pump to be actually reach the blood stream. So therefore, to compensate that, we applied 2 consecutive actuations at the scheduled times. As you could see with the MDP 1A, 2A groups, the maximum concentrations in the blood plasma, again, reached to the level similar to the subcutaneous injection groups. And this pump was able to, with replenishment function. So we replenished this pump with a fresh insulin solution at 30 days. And then we performed, again, the actuation experiments. And then as you could see, the maximum concentration that we observed were, again, similar to the ones that we could observe before replenishment. Therefore, this pump can be envisioned to be used semi-permanently by doing just simple replenishment procedures. And as a pharmacodynamics, we measure the blood glucose levels. As you can see, control groups show very high blood glucose levels. This is type 1 diabetic animal models. And subcutaneous injection groups, in this case, we only measure the minimum concentrations we could observe after administration of insulin. And then those profiles were quite similar to the ones that we could observe with the pump groups. And however, after 11 days, with just one-time actuation, this was not quite satisfactory because, again, insulin could not reach to the bloodstream because of the fibrotic capsule formation around the pump. To compensate that, we simply increased the number of actuations by applying 2 times of magnets from the outside of the body. And the blood glucose levels, again, dropped to the similarly to the ones that you could observe from the subcutaneous injection groups. And after replenishment, again, those drop level of the glucose levels were quite similar to the ones that we could observe before. Therefore, again, this functions properly even after replenishment. We performed the histopathological analysis with the tissues that were biopsied at the end point of experiments. Again, we stained them and then delivered them to the pathologists in our hospital. And they again said that they could not observe any abnormal or bad reactions. Therefore, we could say that the device herein was well designed and then properly functioned without safety problems. And as I said, the fibrotic capsules were indeed formed around the implanted pump. However, what we observed here is that this fibrotic capsule thickness does not increase further, even after 30 days, and then this profile was similar, which means that after implantation, if we wait a certain period of time until the capsule is formed and then stabilized, then we don't expect any further increase of barrier of the drug diffusion from the pump, infused pump, to the bloodstream. Therefore, this could be reproducible, so that as long as we fit the dose and the number of actuations, then the pump will do the same operations afterwards. For this pump can be actuated with magnetic field, so therefore we could design the controller to be outside of the body. Well, the controller can contain, in this case, the outside controller could contain battery because it can be easily exchanged. And then we can make this work automatically by electromagnet to be attached at the outside of the body. And with this controller, we can make this communicate with the mobile app. With a mobile app, we could also add some more safety functions so that like overdose or any mistaken insulin delivery does not occur. So we can prevent them. So this is the design, whole package design that we envision, and actually we developed some prototypes with this pump to be implanted inside of the body. So for conclusion, we developed a simple assembly implantable infusion pump for delivery of insulin. And then this could be actuated with a non-invasive magnetic field, and then the pharmacokinetic and pharmacodynamic profiles were quite similar to the ones that we could observe with that of a conventional needle injections. Therefore, we envisioned that this technology could be an alternative to avoid any multiple daily needle injections. This is the funding source. And then Dr. Seung-ho Lee, now at Koku Institute at MIT, he was the first author of our work here. And then Professor Lee Chol, he is a pathologist in a hospital to evaluate the tissues that we biopsied. And then Professor Yong-min Cho, he's at the Department of Internal Medicine. He's a clinician to help and then to do the research together with. And Professor Jin-ho Choi, he co-works with us to make the stimulus responsive membrane. And Professor Hee-Chang Kim in our department, he works together with us to make this mobile app, and then the device to be used at the outside of the body for a controller. And thank you for your attention.
Hello. Thank you for attending this lecture. We're going to talk about a mechanism that can be used for the dynamic regulation of tight junctions within the intestinal epithelia to facilitate the oral uptake of peptides. As we think about the barrier properties of the intestinal epithelium, we think about two major routes by which we could facilitate the transport of a peptide therapeutic. And one is between adjacent epithelial cells, and the second one is through epithelial cells through vesicular trafficking. Today we're going to talk about the paracellular route, that is between adjacent epithelial cells. It is not the predominant surface area component, that is, you can see on the right-hand side of this slide. You can see that there is a limited amount of surface area here between adjacent cells. So you're never going to get a very large amount of material across, but you're going to get a reasonable amount if you position the material adjacent to those surfaces and if it is open sufficiently long to allow the uptake. Now, what we're going to talk about today also is the fact that there are dynamic modulation pathways by which the tight junction, or TJ, can be regulated. We know that these cells, the epithelial cells, are constantly turning over. That is, the tight junctions have to be dismantled. New cells have to come in. New established tight junctions have to form. And also we know that the epithelial cells have to be able to open and close these tight junctions to allow the transport of entire cells, neutrophils, lymphocytes, things like that. We also know that cytokines such as interferon alpha, interferon gamma, TNF alpha, can dynamically regulate these tight junctions and their properties under conditions of inflammation. What we're going to talk about today is the last thing on this list, which is the fact that nutrients have the capacity to dynamically open and close tight junctions. And in particular, we're going to look at essential amino acids and also glucose, the two things that actually dynamically and structurally regulate these tight junctions. Now, when you think about how permeation enhancers work, in general, all the permeation enhancers that are out there that are currently being examined and have been tested and actually have some use in the clinic right now really were identified empirically. They really weren't identified through a strategy of mechanism of action. They were simply identified through screens, and then with time, a variety of studies helped facilitate our understanding of how they might actually work. But still, to this day, we don't know exactly how they work. What we're going to talk about today is an approach where the mechanism of action is what drives the identification of the permeation enhancer, which is opposite to what people have done in the past. So if we think about how tight junctions can regulate, there was some work done by John Pappenheimer at Harvard Medical School years ago. And what he identified was that when you think about how these essential amino acids move across epithelia, you see a transcellular pathway. And so you're seeing things moving from the apical to the basal side of the cells through restricted systems that regulate the uptake and transport. And these restricted systems are usually sodium-dependent processes. So sodium-dependent glucose, sodium-dependent essential amino acid transport across the epithelia. But under supraphysiologic levels, when you have a very large amount of material, nutrient material in the lumen, what he saw was that you saw not only this transcellular transport, you saw an additional route, an additional acceleration of material across the epithelia, and he coincided that with the idea that you had opened up a paracellular route. You had actually facilitated the uptake by this paracellular route, and what you were seeing was the combination of paracellular and transcellular uptake. Through work with actually someone else at Harvard, Jim Madera and Jerry Turner, who is now actually another professor at Harvard Medical School, this combination of us, we sat and looked at this prospect and really tried to understand what was going on physiologically. And if you think about the way that the tight junction is regulated, it turns out that it is in two forms under these conditions of nutrient stimulation. It can be in a closed form and an open form. And a myosin light chain is what regulates the structure between the closed and open form. So there's a kinase called myosin light chain kinase, which phosphorylates the myosin light chain Under those conditions, the tight junction opens, and when it's dephosphorylated through myosin light chain phosphatase activity, the tight junction closes. Now, under normal conditions, the tight junction is closed. That keeps the unrestricted uptake of material from the intestinal lumen from entering our bodies. And there are two pathways by which that material is closed, that tight junction is closed, through the actions of myosin light chain phosphatase, that is an MYPT1 and CPI17 regulated control of the myosin light chain phosphatase. But when it decides to open, there's only one pathway that opens it, and that is through a calcium calmodulin uptake or calcium activation of the myosin light chain kinase. Now, we went through and initially demonstrated this whole concept by designing a peptide called PIK, P-I-K, which is a permeable inhibitor of kinase. That is a small peptide which could go inside cells, could manipulate the kinase at the tight junction within the tight junctions, within the cell, the cytoplasm, and regulate this and show that we could close open tight junctions in the status of inflammation. Now, opposite to that, you would say, well, then you could make peptides which would then regulate the CPI17 or MYPT1 function, these PIP, or permeable inhibitor of phosphatase peptides. And you should be able to do the same thing. You should be able to now open a closed tight junction because the kinase would, under basal conditions, continue to drive this opening of the tight junction. And so we actually explored this and indeed demonstrated that the inhibitor of the kinase did close the open tight junctions, and that with these peptides that I'll talk about today, which were defined to actually regulate the phosphatase, could open a closed tight junction. So how did we identify these peptides? So we identified these peptides by looking at the crystal structure of the elements involved. And we aligned these peptides with these interactive components, the CPI17 or the MYPT1, with the phosphatase, and looked for specific regions by which we could disrupt those contact interactions, designing peptides which might facilitate that function. We identified the peptides we wanted in these various sites. We then took those peptides and generated small structures out of them with positively charged amino acids on either side so that they become membrane permeant. That was a very important point because remember, all this is happening inside the cytoplasm of the cell at the inside surface of the tight junction structures. So in an overview fashion, we can think of the PP1, the phosphatase we're talking about, as being regulated by MYPT1 and the CPI17. And that the two different classes of peptides we generated, the PIP250 was to block MYPT1 interaction, or the PIP640 was to block the CPI17 interaction, in both cases increasing the phosphorylation state of myosin light chain, decreasing the non-phosphorylated form. And in doing so, we should theoretically be opening the tight junctions, just as an enhancement of the kinase would do. These peptides were then after design, were then flipped in the opposite direction because within the intestine, the peptides are not going to last very long in the peptidase-rich environment of the intestinal lumen. And so what we made is D-amino acid structures with the reverse orientation from the L sequence. And so this allowed us to make stable peptides that could work in the intestine. The positive charged amino acids on both the N and C terminus allowed us to optimize the integration of these peptides into the epithelial cytoplasm, crossing the plasma membrane, getting to where they needed to work. And we could test these in vitro in KECO2 cells, showing that the PIP640 peptide, and here the red lines, was much more active in terms of, on the left side, altering the resistance of these barriers very dynamically. You could add the peptide at time 0. At the blue arrow, you could take the peptide away. You could see the resistance returning. Add the peptide back again at the red arrow. You could see the resistance dropping again. You could see the correlation of the resistance with the properties of transcytosis, or transport of 4-kilodalton dextran. The 250 peptide was slower and more sluggish in terms of its response. That's the gold line. But did work very effectively at later time points in terms of enhancing this change in vitro. We could verify for both the 640 and the 250 peptide. I'm going to focus on the 640 peptide, but we could verify that indeed we can go through selective amino acids, verifying each one to be critical for its properties and behaviors. I'll focus the rest of this talk pretty much on the 640 peptide with that element in mind, but indeed we've done this for both the 640 and the 250 peptide. But we could validate that indeed, we're getting target binding and specificity with specific amino acids within these peptide sequences, and we can correlate that with their properties. And importantly, we could show that indeed the 640 peptide and the 250 peptide bind to the tight junctions. They get inside the cell, they go to the tight junction, they end up participating in the tight junctional function processes. And by changing the properties of this central core of interactions, like in the 640 or the 642 peptide, we lose the capacity for the peptide to interact specifically with those components that drive it to the tight junction, although they still get into the cell. The 643 and the 644 peptide change the functional properties in terms of the specificity and have unique actions at the tight junction or within the cell function behavior. And we can demonstrate this with the idea that as we looked at these analogs and their ability to change not only the resistive properties but the flux, we could show that the 640 peptide pretty much gave us the properties we were looking for, which was not an ablation of the resistance of the pericellular route, but a decrease that was dynamic. And that you could also see when you took the peptide off, it dynamically returned. Importantly, you could see 4,000 molecular weight dextran material transporting, but not larger materials such as the 70 kilodalton dextran, unless we use the 644 peptide as an example, where we have now changed its properties. It no longer functions at the tight junction selectively, and it's changing a different property of the epithelial cells. Under these conditions, you could start to see a disruption of the tight junction and the opening of pericellular permeability to something as large as 70 kilodaltons. Now, what's remarkable about the 640 peptide is the fact that, when you think of an inhibitor, you think of something being downregulated. And when you look at the 640 peptide, the only way that it actually works in this system is when we looked at the tight junctions, the only thing that it seemed to be changing was the tight junction element called Claudin 2. And the only thing that was changed was actually Claudin 2 was upregulated. So the correlation between the increased phosphorylation state of myosin light chain and the increased distribution or content of Claudin 2 at the tight junctions was the only thing that correlated. And this was quite striking in the sense that it really told us that the mechanism of how the PIP640 peptide works was not a destruction of the tight junctions. It was a modification of the tight junctions that was important for their permeability properties. Now, when we looked at the actual upregulation of the Claudin at the tight junctions, indeed, we could see that. We could demonstrate that in vitro, we saw this upregulation of the Claudin 2 levels that correlated exactly with the modification of the tier, the tight junction resisting properties. And that correlation was very dynamic and consistent with the functional properties that we saw for how Claudin 2 was moving around the cells. We could validate the idea that the phosphorylation state of the Claudin 2 was important, and that this whole process was regulating a phosphorylation at serine 208, which allowed the Claudin 2 to actually function at the tight junction and change its functional properties. Now, Claudin 2 is interesting in that it has the behavior of enhancing the pericellular permeability in a perm-selective fashion. Many of the tight junctions are simply there to seal the tight junction, but there's a number of them that actually function to regulate either enhancement of negative or positively charged ions across the tight junction. And in the case of Claudin 2, it's a perm-selective barrier protein for positively charged structures. And so what we could show is we could demonstrate that indeed when we added the 640 peptide, we could actually not only enhance the neutral transport, the neutral dextran transport, but really enhance the positively charged DEAE dextran in comparison to that. When we treated the monolayers with the 640 peptide and compared it to a negatively charged carboxymethylose dextran, you could see that indeed there was comparable transport between the two, no real difference between them. And so this clearly showed that the modification of the 640 peptide on the tight junctions was really a perm selective. It enhanced the enhancement, but perm selectivity enhancement, specifically for the idea of positively charged structures. We could validate that by then taking and doing a knockout of Claudin-2 in these Keiko cell systems in vitro, showing that when you did that, you lost the capacity for this enhanced positively charged upregulation of paracellular permeability, but not the paracellular permeability at all. I mean, you could still see the 640 peptide enhanced paracellular permeability. It just no longer did it in a perm selective fashion. So the modification or manipulation of the paracellular permeability was still there, just simply not in this extensive, positively charged, beneficial way. We could then go on and show that indeed Claudin-2 was also regulated in vivo, and that correlated also with the phosphorylation state of myosin light chain. And so under these conditions, we could, again, discriminate between the tight junctions between the two structures and their ability to actually deliver material across. So, now how does this affect the value of how we think about this in terms of the oral delivery of peptides in terms of therapeutics? And so we could go back and look at insulin, for example, as a first run, and show that indeed these peptides did pretty much what we saw before. So the peptide 250, which we said opened the tight junction quite dramatically, did open the tight junction and keep it open for a longer, more durable time frame. The 640 peptide, in a relation to the 250 peptide, was slower, didn't have as rapid a response in terms of its durability, but it was more dynamic. The idea that you could put an opening of the tight junction and you could get things through that more rapidly with the 640 peptide relative to the 250, but the 250 peptide had a more durable response. And this is just looking at, in this case, blood sugar in response to an oral administration of insulin. And that's all good, but you can get rats to change their blood sugar for a lot of reasons. So we went and characterized exactly what was going on there. We could show that indeed that a fluorescent version of insulin put in the luminal surface. On the left side, you see that sitting at the luminal surface after administration, but you could see that this fluorescent version of the insulin could actually be going down between adjacent epithelial cells once you had the 640 peptide present. And so you're opening the tight junctions, letting it through. And you can correlate that with the phosphorylation state of these tissues in vivo. And the time course of that correlated, again, quite well with this outcome of insulin transport. And indeed, also we could look at the toxicity of this. So if the material wasn't actually, if it was a natural mechanism by which this was all occurring, then we shouldn't see any kind of damage or modification of the epithelial barrier. We weren't seeing that. It was very dynamic. And we could demonstrate that both the 250 peptide and the 640 peptide, under conditions by which they were modifying the barrier function and facilitating the uptake of this insulin material, allowed really no change in the properties or the functional barrier of the epithelium related to things that would suggest a damage to the epithelium. We then went back and looked at the actual levels of insulin in the blood and what you can see. And so in this case, again, comparing the 250 peptide to the 640 peptide, the importance here is that the 250 peptide is slower and probably more imposing, I guess. It gives a larger impact, slightly larger impact than the 640 peptide, which is more rapid and dynamic. And under both conditions, we could measure the amount of insulin we could detect in the portal vein as it entered into the liver, and then we could take systemic exposure levels, that is in the tail vein, to see how much material actually passed through the liver. And so based on these observations, we could actually demonstrate that about between 3 to 4% of our material administered to the intestinal lumen of a rat actually got across the epithelium and into the portal circulation. and that about half of that actually was extracted by the liver before it ended up in the systemic circulation. And that's consistent with, I think, how people think about how insulin regulates at the liver to control blood sugar systemically. So the data suggests that indeed we can get a reasonable amount of this material through. This was not optimized in any way. These were just mixed, injected into an intestine, or put into intestines, measured kind of grossly. But the idea is that a dosage form could actually administer these things in a more efficient way. But the concept is there in terms of the idea of regulating this in a dynamic fashion. Now, importantly, we could also look at this chart selectivity question about the Claudin 2 that I described. Now, the peptide, the 250 peptide, was not shown to have this Claudin 2 specificity, only the 640 peptide. That's another kind of presentation I don't want to go into right now, but the idea is that if we look carefully at the 640 peptide, we could go back and actually do the exact same study where we looked at two molecules. Rather than insulin, we looked at exenatide or calcitonin, which have about the same molecular weight, but very different isoelectric focusing points of 4.9 versus 8.8. So one is negatively charged at the neutral pH of the intestine, one is positively charged at the neutral pH of the intestine. And indeed, the calcitonin transported about twice as efficiently as the exenatide across these systems, just like in the data that I showed you a minute ago, or a second ago, with the insulin. So all of that natural behavior that we've identified with a mechanism, which is exactly the way the body designs and works this, can be utilized for the discrimination or the facilitated uptake of a positively charged peptide relative to a negatively charged peptide. They both move. One just moves more efficiently than the other based on these charge properties and the Claudin 2 specificity of perm selectivity. Now, when we look at this, we could also look at that peptide in vivo and do some more studies where we start to look at actually enhancing these things by putting them in there more efficiently. And I think realistically, people always say, well, how much can you actually get in? Our perception is that we could get almost to 10% bioavailability with some of these molecules. I think that's about the upper limit by which you might be able to achieve under the optimal conditions of organizing this to actually get a pharmaceutically acceptable uptake. And 10% is not an unrealistic thing for a peptide in the size of exenatide or calcitonin. Certainly in our case, the calcitonin would be better because of that negatively charged thing. The positively charged structure, we could probably move that more efficiently. Under the worst conditions, 5% is probably what's realistic for this. Now, pushing that further means that we'd probably have to open the tight junctions more than I'd like, and potentially let in things that we would not like to see. But that is something that pharmaceutically would have to be evaluated. But the concept of opening the tight junctions on this natural mechanism is quite intriguing. Now, one of the things that also happens is that with the 250 peptide, under certain conditions, we can see the tight junctions being dismantled slightly. And that's something we don't want. So under chronic conditions, we think that we could actually start to see some dysregulation of tight junctions. We don't see that with the 640 peptide. So the 250 peptide is something that we would want to avoid under these conditions for a chronic application. So at this point, the 264 peptide is probably, the 640 peptide is probably the one that we're moving forward with in the clinic. So in summary, what we think is going on at the tight junction is that there is this dynamic regulation of materials. There's a natural regulation through Claudin 2 to keep nutrients and things moving through. And the Claudin 2 function probably functions to protect us from endotoxins coming into the body during these superphysiologic nutrient uptake pathway events. Now, under leak conditions where you have inflammatory responses such as TNF-alpha, then everything starts to dismantle and the tight junction starts to be downregulated. And in some cases, that may be what we're seeing with the 250 peptide rather than but the 640 peptide looks more like the pore pathway activity and probably is the mechanism we would want to be using in the clinic. So in summary, we can see that if you were to co-administer one of these peptides with an insulin or a protein or a peptide of some kind, wherever the peptide is entering into the epithelial cells, regulating, disorganizing, or modulating the tight junction function, at those locations you would see this paracellular transport enhancement And as that peptide continued to move through the cells into the circulation, eventually being secreted out of the kidney because they're stable little peptides, they just flush out real quickly, you would see this rapid transient kind of response for enhanced permeability of your peptide. So in summary, what we've talked about is two rationally designed peptides. They were both developed based on a biological principle that was identified related to how the intestinal epithelia regulates in response to nutrient uptake. Both of these peptides seem to regulate in slightly different ways the tight junction properties. And because of that, we can think about using them in discrete ways in terms of enhancing paracellular permeability of small peptide therapeutics. So with that, I'd like to thank you for your time. I hope this was informative, and hopefully we can take some questions and discuss this. Bye-bye.
Okay, I want to thank the organizers for this opportunity to present today our data. And my lab is studying how to manipulate cellular function in order to generate novel therapeutic approach to treat mainly leukocytes implicated diseases. And this is a huge field that include, blood cancer, inflammation, and viral infection. We are quite occupied in all those bluish diseases. So our lab is divided into two major groups: the delivery people that generate lipids, ionizable lipids, novel ionizable lipids, do some protein engineering and generate monoclonal antibodies, as well as doing drug discovery, exome transcriptome analysis. In an ideal world, we want to combine those two into one nano vehicle that will tailor-made everything in one vehicle and will target specifically to specific cell types. So we are from the field of RNA, and therefore we have to copy from somebody who's smarter. We have learned, not in the easy way, that RNA delivery and gene manipulation in leukocytes is a huge challenge. Not only you need to do everything related to drug delivery, efficient encapsulation, specificity, evading clearance mechanism, and avoiding toxicity and immune activation. And of course, the carrier must internalize because we are talking about RNA payloads and endosomal escape. But moreover, if you look at these guys, these are schematic illustration of lymphocytes and naive lymphocytes, and you can appreciate the majority is a nucleus. Therefore, you have to really sneak in very quietly without being identified. And if it's an activated one, you have a little bit more cytoplasm, maybe 25% of the cell's cytoplasm here. And there are lots of sensors inside, so we have to be extremely, extremely sensitive and stealth. So Alnylam made a huge dramatic jump by approving patisiran or Onpattro, which is the first siRNA in lipid-based nanoparticle that goes to the liver, and it's a rare genetic disease, and it's amazing. I'm going to tell you a few stories, starting with inflammatory bowel diseases and going all the way to cancer. And in inflammatory bowel diseases, we're interested a few years ago to understand why immune cells, both in Crohn's disease and ulcerative colitis, have this gene, cyclin D1, and I would say aberrantly upregulated in leukocytes. We wanted to understand if it's contributed to the pathogenesis of inflammation, and if so, can it serve as a therapeutic target? And we set up a very naive strategy to target gut mononuclear leukocytes and selectively silence cyclin D1 in experimental colitis model. But this was the first systemic cell-specific delivery of RNAi payloads to immune cells. And we started with a multilamellar vesicle. We extruded it to a unilamellar. We put hyaluronic acid or hyaluronan as a scaffold and as a replacement to polyethylene glycol. And on the surface, we generated some monoclonal antibodies against the beta-7 integrin. Beta-7 integrin is a homing receptor that has been upregulated when there is inflammatory bowel disease, and it's being upregulated from the circulation on leukocytes that home to the gut. So we thought that maybe by blocking it at the same time, the ability to penetrate with cyclin D1 siRNAs and silence this gene might be beneficial, because there are a few papers back then showing that in IBD specifically, there is a direct correlation between manipulation of, homing of leukocytes into the gut, as well as their ability to proliferate. So there was a direct connection between proliferation and migration. And we thought maybe we can block this proliferation, and hopefully the migration will be decreased. So we set up this strategy. We showed that on the shear flow condition, those anti-beta 7 particles penetrate very nicely into lymphocytes, primary lymphocytes, where you have an isotype control on the surface, it will not penetrate, and siRNA per se cannot penetrate into those cells. We also did a hallmark experiment, a killer experiment, by using a beta 7 knockout splenocytes. Here you don't see basically anything. It's a black image. There is no receptor. They're not going into the cells. The reason it was published in this journal was that we also identified new mechanism by which cyclin D1 is controlling some of the pro-inflammatory cytokines, such as TNF-alpha, IL-12P40, also IL-23, which share the P40 units, IL-2, interferon gamma, etc. But it doesn't change the IL-4 and IL-10. So basically, there is a very gentle balance between inflammation and pro-inflammation. It doesn't really touch this. So we show that this has a therapeutic value. And you can see here, for example, in a DSS colitis model, how leukocytes infiltrate and destroy the structure, where when we use this anti-beta 7 integrin with cyclin D1, it has actually almost an intact structure. So what happens with this? This was translated, unfortunately, not in the form of a particle, but in the form of the anti-beta 7 antibody, and entrolizumab is now in phase 3 clinical trials. Hopefully, we will know by the end of the year if this is an approved drug or not. As you all know, complexity of human diseases is really a big issue. And we are very much interested to target our payload to specific cell types. We are predominantly interested in siRNA and messenger RNA, also self-amplifying RNAs and circular RNAs. So we use microfluidics, mostly the nanoassembler, which is a very elegant way to create formulation from lipids and different oligos, mRNAs, etc. And you can get very stable particle that looks like this in cryo-temp that have all the parameters that we are interested in. So as I mentioned, Al-Nyland did a fantastic job, and Peter Kulis with his MC3, the D-lim MC3 DMA lipids that we actually learned a lot from them. These are non-degradable ionizable lipids that accumulate in the liver, and Dr. Srinivas Ramishetty from my lab basically, based on those ionizable lipids, designed new lipids by changing the cationic head groups, the linkers, and the hydrophobic tails, and we came up with about 60 lipid families. And so we tested them and recently published it in this journal. And again, the same strategy. We basically package those nucleic acid with those lipids in microfluidic mixing devices and get stable particles. And this is an example of different lipids that gives you different diameters, different polydispersity index, and different zeta potential. And you can also see how nice they look. So until now, I actually talk only about this size. So you have very stable particle. The problem starts when you want to give them direction, target them to specific cell types using not a natural ligand, but an antibody. To do so, you have to reduce the antibody, put a pegmalamide on the surface, and then ideal world, you will get something like this after removal of unbound antibody with a gel filtration chromatography. Is this real? Well, we don't believe so. So as you probably know, chemical conjugation has a lot of issues. So it really depends on the conjugation, but if you radiolabel the antibody, you will get the efficiencies ranging from 0.1% to maybe 10%. This is the efficiency that we get, not more than this. Antibodies are not alike. Some of them has glycosylation. It's very hard really to orient them into the right area. And if you don't orient them, what will happen is that you'll have FC exposure with very poor binding. So if you inject those creatures into the bloodstream, they will be taken out very quickly by cells having FC receptors. So we decided to move from chemical conjugation to what we call a universal linker strategy, going back from chemistry to biology. And here we generated what we call affinity approach, basically creating an antibody against the antibody, an antibody against the Fc portion of the antibody. Therefore, you just need one conjugation, tiny amount of antibody, and it will orient itself very well and in a very efficient manner. You also then have protection from the Fc. And if you imagine that you're a particle, disregard the ears very smoothly, and you want to avoid mononuclear phagocytes, usually macrophage, dendritic cells, monocytes, we usually have a very simple trick. We pegylate those particles. This is a pegylated version. And if I want to explain to you how important it is to have a good design of orientation, you can look at these two different knives. One has two blades. It's very hard to cut anything with this. And one have a few blades oriented. So you can see the mission here is much easier. So behind this approach were two former graduate students of mine, Ranit Kedmi, now immunologist at NYU, and Ofar Varga, now is cell biologist at Leuven University in Belgium. And basically, we came up with this approach by basically having an antibody against the Fc portion. Then you just need to mix them, and they will go to the right direction. Okay, I love this image because it gives you the opportunity to really target specific cell types, one platform, different antibody, different cells. And what happened is we started with the complete IgG. We created a single chain based on this IgG, and we found a sequence that could be lipidated in one transcript. So we have a messenger RNA that could be a transcript from a single chain to a lipidated single chain, with the help of Professor Itai Benar, who is a protein engineer at Tel Aviv University, and Mark Belke, who is a great nucleic acid chemist. So a few years ago, we published this, basically taking the nucleic acid and the lipids with the microfluidics, and then you have a stable particles. Then in bacteria, you produce those single chain lipidated version. You put a little bit of cholesterol, you mix them again, you have a post-insertion very effectively, and then you put an antibody on the surface, and this is what's basically your product is. It's very efficient. It has very good affinity around 0.4 nanomolar, and this is the construct that we designed. So basically, this is the single chain. This is the lipidated version. It goes into the particle, and you have a platform. And this is a great platform because you just mix different antibodies and go to different directions. For example, when we put an anti-beta 7 antibody on the surface, we expect that it will go to all leukocyte subsets. When we put anti-CD3, it will go only to T cells, anti-CD4 to a subset of T cells, anti-CD25 to a subset of CD4. And this is exactly the experiment that we have done here. So we put Cy5 label siRNA inside and basically injected into mice. And 1 hour later, we took splenocytes. And you can see that if you have an anti-beta 7 antibody on the surface, as expected, it will go to all leukocyte subsets. Anti-CD3 will go only to T cells, where anti-CD4 will go only to CD4 cells. So CD25 expressing and CD4. Anti-CD25 will go only to CD25 expressing cells. Can we silence something? So we got from Alnylam, we had a long-standing collaboration with them. We got anti-siRNA against CD45. We entrapped it inside. Then we put a CD4 antibody that will go only to CD4 cells, or an anti-beta 7 that will go to all cells, for example, CD4 and CD8 cells. And this was the experimental design. We put here the SI-CD45 or SI control. We put an antibody or an isotype control, again, the CD4 or beta 7 integrin. We inject into mice. We wait for 5 days. We isolate splenocytes and we fax them. And this is basically what we got. This is the expression of CD45. If you look at those two cell population, if you have an anti-beta 7 antibody on the surface, it will silence both types of cells. If you have a CD4, it will only silence the CD4 cells and will not touch the CD8. So what we have done, we have used two animal models, one for colitis and one for mantle cell lymphoma. The colitis model here, we use inflammatory leukocytes as the receptor targets is Ly6E high, and the target gene was TNF alpha. And this experiment was done blinded, as you can see here, in all parameters, colonoscopy, markers, relative IL-6 level, TNF level, colon length, etc., etc. You can see that the groups that got TNF anti-Ly6C with siRNA against TNF got the most effective therapy. Mantle cell lymphoma, here we use an anti-beta-1 integrin or anti-CD29, and pololykinase 1, PLK1 is the target gene. This is a rare disease, a very aggressive form of a B-cell non-Hodgkin's lymphoma, where about 6% of the non-Hodgkin's lymphoma is still considered to be incurable, although there is treatment with combination of rituximab and anti-CD20 and chemotherapy, or the new BTK inhibitors. It's still the poorest among the B-cell lymphomas. Okay. And so we developed a few years ago an animal model for this, and we show that we can implant cells, either human cells or cell lines in mice, and they will develop inside the bone those tumors. And within 24 to 30 days, those animals will die. So this is an exceptionally good model to study whatever you have. And looking at the experiment we have done, you can appreciate that a few injections, we can extend the survival dramatically. Okay, also from, if you look at the median survival. Then today, it's an interesting day because Pfizer had just announced that they have exceptionally good results with vaccines for COVID-19. It's based on an mRNA. So I'll tell you a little bit about what we have done with an mRNA, and we are the first to show systemic delivery of mRNA to specific cell types. And so Nofar from my group basically utilized exactly the same system with which we call the ASSET system, and show that she can put an mRNA inside. In order to do a quick experiment, here we chose again a colitis model. Target cells will be inflammatory leukocytes, and the target gene to upregulate will be IL-10. IL-10 is an anti-inflammatory cytokine. It's very important. And the idea behind this was that we may be able to create a factory, cells that produce for us IL-10 in the vicinity area of the microenvironment of the gut, and they will be affecting all the gut environment for basically creating an anti-inflammatory situation. So in order to start, we put luciferase siRNA with an anti-Ly6C. You can appreciate that you can sort the cells that are Ly6C minus or positive, and you can see the difference between a targeted nanoparticle that have luciferase mRNA compared to an isotype control. So if they're Ly6C negative, there is basically no difference. But if they're Ly6C positive, you can see 2.5 log magnitudes here. So this is substantial. This is what we call the targeting index. And so the expression of luciferase that you can see here is in the intestine and the spleen and the liver, and it's actually a good marker to do some in vivo work. So this is our approach. You inject systemically, you target those inflammatory leukocytes, they're entering the cells, you're releasing your payload, which is an mRNA that will be recognized and translated through a ribosome, and this IL-10 will be now secreted and will affect and change the gut microenvironment. This is exactly what happened after 3 IV injections of this modified mRNA. So in any parameter we look, IL-10 level, IL-6 level, you can see here, nice reduction, almost to a level of healthy. Colon TNF, exactly the same here, okay? All the controls are here, as well as body weight changes, colon length, and histology. We also looked at safety profile by injecting those particles and looking at blood counts, biochemistry, liver histology, and in all of them, it looks very promising. We also want to use this tool as an in vivo drug discovery, and we had a bet on IRF8. IRF8 is a very interesting target, interferon regulatory factor 8. Basically, it can do many things. It can differentiate towards mononuclear phagocyte lineage. It can basically upregulate the expression of TNF. and it's also can change commitment from dendritic cells. So basically, the idea was to inhibit this by using siRNAs. Exactly the same concept. Anti-Ly6E, inflammatory leukocytes as a target. Here with siRNA against RF8, 3 IV doses of 1.5 mg siRNA per kg body, looking at weight, colon length, colonoscopy, and cytokines in colitis model. And basically, in on-parameter, we also see that these guys have a lot of potential. So this is what we think happens as a model here. We think it's a novel therapeutic target, IRF8 in IBD. And again, the idea is that these guys target inflammatory leukocytes entering and reduce the IRF8 expression. If they reduce the IRF8 expression, they will influence and reduce TNF, IL-6, IL-1 beta, and IL-12P40, which shares this unit with IL-23. And therefore, this is what we believe. This was the cover last year of a Journal of Control release. A few more slides just to show you a really quick, nice paper that was published last night. It's a therapeutic genome editing in cancer. Exactly the same system. That is the same idea. Here we entrap mRNA for Cas9 and single guide RNA. This is actually the first ever strategy for cell-specific delivery of basically CRISPR nanoparticles, and a very efficient one, as I'm going to share with you. We can either put nothing, or we can put an antibody, and the same idea is anti-EGF. And just to show you very quickly an example in a very aggressive ovarian cancer model, that we can see 80% genome editing, about 80% change in survival, which is really, really dramatic here. After 2 injections, we have also brain glioma tumor, intra-tumor injection into the tumor bed in a stereotactic instrumentation, 1 single dose and follow-up of 60 days, and we get really amazing results. So I urge you to read this. Just to conclude, we have developed some safe and seems to be non-toxic formulation. We've shown this in IBD models. We've shown this in cancer models, and also very recently we're going to show some data on rare genetic disease. It gives you endless opportunity for precise gene manipulation. And again, I haven't had the time to talk about fast internalization and burst release, which is really important. Avoiding more nuclear phagocytes, high specificity and functionality, and of course, better RNA-based drugs. At the end, I just want to thank my group of really outstanding scientists coming from diverse fields, including biology, chemistry, medicine, engineering, material scientists, etc., and the funding agencies and companies that work with us. And I want really to thank you all for listening. I'm going to end up with a very short movie.
In inflammatory disorders, the sick cells in our body are our own white blood cells. Only instead of defending the body, they attack it. The best way to fight the complexity of human diseases is to deliver the therapeutic payload directly to the sick cells. A promising technology for combating diseases uses special lipid nanoparticles. These nanoparticles contain RNA molecules that can change the way cells produce proteins and can modulate cells' function. The nanoparticles are supposed to deliver the RNA directly to the sick cells. The problem with today's technology is that the nanoparticles hardly reach immune cells, but rather affect other healthy cells instead. We at Pear Lab have developed a new platform called ASSET that can send RNA nanoparticles selectively to the correct cells. While existing methods of connecting nanoparticles to antibodies are extremely complicated, Pear Lab's method is a matter of simply mixing 2 solutions. And the nanoparticles are decorated with an ASSET linker, which then binds the antibodies. Each type of antibody knows to attach to a very specific type of cell. So when they decorate the nanoparticle, they will direct it only to the targeted cells. This method means releasing the RNA right where we want it, affecting only the right cells and saving the body from harmful side effects. Asset nanoparticles are a novel strategy to fight against inflammatory diseases and even cancer. And this time, it's personalized.
Okay, thank you so much.
Now this floor is open for questions and answers. So I got the questions from the floor, and I welcome Professor Sarmiento and Mrisny and the peers for all great the lectures. And I'd like to ask the questions to Bruno first. So the question is that the first question is, when albumin is taken up by the cells via the FcRn-mediated endocytosis, they might be recycled to the cell surface. So how do you think about the possibility that your engineered the nanoparticle could be recycled back into the cell surfaces or degraded in endosome rather than transcytosis?
Yeah, that's a good question, and that's a question that we addressed before. So in terms of recycling, we also observe some recycling of the particles after the internalization. So we quantify it, but luckily the amount of recycling nanoparticles is much less compared to the amount of nanoparticles that were transcytosed. And regarding the potential degradation of the albumin, we also observe that by the fact that albumin is bound, is linked, covalently linked to the nanoparticle, it is less prone to be degraded compared to the free albumin. So what you observe is that when you quantify the amount of albumin that we, on an in vitro test, of course, when you quantify the amount of albumin that is recycled and that is transcytosed, we have just negligent amounts of albumin that disappears, or mean that will degrade. So we believe that the free albumin will be degraded, some of it, but not as it is bound to the nanoparticle. About the recycling, so this is a fact, and there's a drawback that is associated with this receptor, but this is common for most of the receptors that do recycling process. So we know that the process is not completely in one direction. So we have some recycling of the particles that are bound, but still the bioavailability deserves to explore more this route. So yeah, it's a compromise between the transcytosis and the recycling, but still the transcytosis wins this fight, I guess. Yeah.
All right, thank you. And the second question is that, Bruno, you mainly talked about the application of FcRn-mediated transcytosis for oral delivery system design. And the question is there any other application other than diabetes? What do you think would be the next disease model to apply your concept?
Okay, so this is not exclusive for the oral administration. We know that this transcytosis mediated mechanism is also explored for the lung delivery, for instance, in terms of mucosal delivery, and with very good results. But as far as the levels of the FcRn are expressed independent on the disease that we want to target, I believe that the platform can be adapted for many other drugs. So this is a quite flexible platform. It is, the FcRn expression is not dependent, for instance, in the case of diabetes. Something that I see that could be more advantageous is the exploitation of diseases that mostly affect young adults or of childs, because the level of expression of FcRn is higher for the youngers compared to the older people. This is a fact. So in terms of saturation of the absorption process, probably we'll get saturation sooner in elder people compared to the younger ones. So if you want to explore this route for diseases that are more common in young people, I believe that it's an advantage, but not specific for diabetes for sure. For many other diseases, yeah.
All right, thank you. The third question is that have you ever tried to deliver this FcRn system by systemic application? If so, what would be the distribution level of FcRn in the blood vessel? Is it possible that they are also taken up by blood vessel epithelial cell and regurgitate? Yeah.
Yeah. We never tested because our aim is to have mucosal delivery of biopharmaceuticals, so this is the topic of the group. But I know that many others that are trying to explore not the same concept in terms of functionalized nanoparticles, but in terms of fusion proteins, where they can couple albumin with many other active molecules. And the idea is to improve or increase the half-life of the drugs, precisely because they take advantage of the recycling process at the endothelial cells. So it's a very well-known mechanism of increasing the half-life when you administer intravenously a drug albumin conjugate. And with this strategy, so you have a clever system that increases the half-life of your drug, and overall, I mean, all the advantages that are related with increasing of the half-life. Not necessarily nanoparticles, but albumin drug conjugate, which I believe it's more relevant from the physiological point of view.
All right, thank you, Bruno. So I will now ask questions to the second speaker. Yeah, Bruno, I think that is about it for your part. And Professor Yongbin Choi, is there any possibility that your device can induce some inflammation? What's your opinion about the safety aspect of your device?
Okay.
Thank you for the question. And our device is kind of non-degradable and has got some volume. So when it's implanted, we cannot fully avoid inflammation. Anyway, inflammation occurs because anyway the healing has to occur to actually make the device to be stable inside of the body. But however, after acute inflammation, what we found was there's some kind of fibroid capsule formation to actually isolate the device from the body. And that was okay in an aspect of the device itself. However, as I showed in some of our results, that because of this capsule formation, the drug exposure to the systemic level became different as the inflammation is progressed. However, what he found was the capsule thickness was kind of stabilized afterwards. So if you wait after the implantation of such kind of device, and then when the capsule is stabilized, then there is no more inflammation, and then drug can be released in a reproducible manner to the systemic level. That's our belief.
Okay, so it is actually, second question is that it is basically implant system. Does it mean that you have to take it out again by surgery?
Oh, yes, that's a good question. That's why we did not want to use any battery or electronic circuit inside. Rather than we would like to make them operate through some kind of anionic radiation or magnetic force, especially for the insulin delivery, it's for chronic therapy. So what we envision is that the device needs to stay there for a long time with a replenishment ability. Therefore, we don't need to explant the device because there's no battery inside.
All right. The third question is, what is the thermal stability of insulin? Because you use the NIR to increase the heat. So what if the insulin is not stable and denatured by heat? What do you think about it?
For the first device, when we used the NIL, the reservoir was not filled with a liquid formulation, rather it was filled with a dry formulation. And we did not try with the insulin for that device. It was tested with growth hormone. And what he found was the growth hormone was stable as the dry formulation for the period of implantation, and longer than that when the device itself was incubated at 37 degrees Celsius. I think it was possible because it was dry formulation. And for the second device, we also tested the liquid formulation insulin to stay at 37 degrees Celsius for more than 60 days. And then we found that the activity and then concentration of insulin did not change. And when we took out the insulin from that testing device and then put that into, injected into the animal with the diabetic 1 model, we found that the glucose level dropped similarly as shown with the fresh insulin. So we believe that as long as we can make that formulation with the drug stability, then probably we can resolve many issues with that.
I see. All right, I think that explains all the questions. Now I'll move to Professor Randy Milsney for first. Thank you for the wonderful presentation. And the first question is that what do you think would be the preferred route of your peptide 640 and 250? What is your preferred administration route?
Well, thank you for, again, the invitation to participate in this meeting. I didn't get a chance to say that earlier. Very nice and really wonderful to see all the people that are here. So it's like an old return to friendly faces. So preferred route is all oral here. The whole pathway is focused on a nutrient-driven system that really applies only to the intestinal epithelium. Things are similar in other epithelial of the body, but the mechanism specifically that we're addressing is in the intestine.
All right, so in relation to the first question, the second question is that, so do you expect that when you try different routes like a nasal delivery, do you think the enhancer effect would be changing? Yeah.
It does, yes. It does work. It works in those other places, but the reason that we're interested in the oral system is because the oral system does dynamically open and close every time you have a meal. Your airways don't open and close dynamically in the same way. They still have the same mechanisms, but they don't do it in this kind of continual, very structured and very controlled fashion.
All right. The third question is that you showed that your peptide 640 works for Claudin and your peptide 250 works for occludin. So because they are working on two different proteins, have you ever tried to combine two peptide systems for synergistic effect? That's another question.
That's the very obvious one, yeah. So what we've looked at is we have explored that. You do get sort of a synergistic effect. It's not really how the body uses these pathways, though. And so what we're trying to do is align as best we can with how Mother Nature sort of stabilized the system. The 250 peptide and the pathway it modulates really kind of describe inflammatory systems and how neutrophils and things move through that environment. The 640 peptide much more emulates the way that nutrients dynamically open and close the tight junctions for enhanced nutrient uptake. If you're going to deliver things chronically, then you probably want to use the 640 peptide. If you want to use them and deliver slightly larger molecules just on occasion, then the 250 peptide could theoretically be useful without too much toxicity associated with it. So that's sort of the way we're thinking about them.
I see. So what do you think is the tight junction opening time of those peptides? Because that kind of duration is also a crucial issue. Okay.
So the 640 peptide is very dynamic. We see it within half a minute that we start to see what looks to be opening the system. It's a very quick thing. And that probably a lot of that time is for the peptide to actually get into the cell, crossing the membrane, getting into the thing, organizing itself at the tight junctions where it acts. And then we see as the concentration of that material decreases inside the cell, again, within a minute or so, that tight junction is closing up again. So as long as the tight junction is being modulated by the 640 peptide, it will stay open. And so as long as the 640 peptide keeps coming in from the surface, it'll keep that concentration within the cell reasonably high and keep the tight junction open. So that's simply a process of the pharmaceutics of how you make an oral dosage form that delivers that to the luminal surface. The 250 peptide will open it and stay longer. And so there we see things opening up within about 5 minutes or so. And then it starts to stay open maybe for 15 or 20 minutes at the end as it starts to close up and reseal. So again, one is quite dynamic. One is a little slower in its actions in vivo.
All right. Maybe the last question for you, Randy. What do you think is the pH stability of your peptide? Because anyway, if you give it by oral, they have to pass through the stomach with low pH. Have you ever tested?
Yeah, so actually the dosage form would actually take care of all of that, because it's not so much the peptide. The peptide is actually made all out of D amino acids. It's actually quite stable against peptidases. It's the cargo that you're delivering with it is the problem. So if you're delivering, salmon calcitone or something like that, it's going to have the bigger problem when it comes to stability as it passes through the intestine. So it's all about making oral dosage forms that are protected in the stomach, that open at certain regions of the small intestine to allow both of the components, the peptide and the cargo of interest, to be released at the same location adjacent to tight junctions that they can open.
I see. Thank you so much. I think that is about it for you, Professor Milsny. And I'll move on to Professor Dampier for your part. So it'll be really interesting to develop your mRNA vaccine. So the first question is that currently the mass media says that mRNA vaccine from Pfizer are suffering from the mass production and the transportation issue. And the question is, what is your opinion for that kind of stability? And is there any other strategies to overcome that kind of transportation issue to improve the stability? That is the first question. Dan.
Okay, first I'd like to thank you again for inviting me, and also to see very good friends and colleague here. So, that was already great to see Randy, and Bruno I saw yesterday as well, but wow. Okay, thanks. And to your question, I think that, there are two factors. One is the mRNA for the vaccines. The other one is the lipids. I'm a bit biased because we are, and it's in the public domain, we are collaborating with BioNTech that licensed some of our lipids a few years ago for different reasons. Was supposed to go to cancer program and to seasonal flu vaccines with Pfizer, but it happened. So I think that there are two issues. One is the mRNA, the other one is the lipids. Okay, so you can modify, mRNA is very sensitive, also the lipids are sensitive. Until recently, the ionizable lipid that I've talked to, which is the same that Alnylam was using, that came from Peter Coulis, is actually very stable. And also in terms of it's probably stable because it's non-degradable. I think that when you have a degradable lipids, and these are all the new types of lipids that are degradable, both in Moderna's and in Pfizer-BioNTech are degradable, and also in Arcutus and other companies. So I think that one issue is the stability of the lipids. The other one is the stability of the mRNA and how many different mRNAs are in the formulation. So having said that, there are two factors, and you can see that there is also a game between the companies saying who is better and who has, better cooling system. I would not surprise if we will hear next week that somebody come up and say, I have a formulation that is room temperature stable for at least 2 months, okay? So I don't know. I think it's a little bit probably early to say many things about the stability. I think it's also very early to say what is the percent efficiency of the virus, of the vaccines. I think that people have to be aware, and again, I'm talking only on my behalf and not behalf of any company here. People have to be aware of the fact that, for example, if you are positive for corona and you live with a house of people, some of them, for some reason that we don't really understand, will not be infected. And therefore, all the percentage of 90 patients or 100 patients is a bit obscure here. So I think we have to be careful. Probably they will get some kind of approval for at least, I don't know, a year. We'll have more data. When you'll have data for 100,000 people, then we can talk.
All right, thank you.
Long answer for a very short question.
Yeah, the students will like it. The second part is that you showed the cartoon that the mRNA or some nucleic acid are encapsulated inside of your nanoparticle. How you can exclude the possibility that nucleic acid also kind of adhere on the external part of your nanoparticle?
Okay, that's a good question. So it's actually very simple. We are using microfluidic systems, and basically, without doing a lot of publicity for the specific company in Vancouver, basically you have two inlets. One is your lipids, or the other one, or a mixture of lipids and a low pH in ethanol, and the other one is then your nucleic acid. And in the form of RNA, and it could be as long as 17.5 kilobase or very, really, really, really large self-amplifying RNA, there is no way that, you know, if you do it in a low pH, your lipids will be highly positively charged. And you bring together something very negative and very positive. I don't know if you can see my hands. That's the problem with this. But basically, you're bringing them together and you mix them in a very fast reaction and very, very small volumes. And basically, the chances that now you will have something outside is almost zero, almost zero. And, you know, luckily, we have had to prove it in 2014. But before us, Al-Nyland showed it many, many times that there is nothing stick from outside. You can use RNases. Nothing is there. So almost the encapsulation is somewhere between 97% to 100% or 99.99. Very efficient. It depends, of course, on your lipids. One thing people need to remember that afterwards, after this particle stability, you have to change or you have to dialyze your particle to change the pH because they were produced in a mildly acidic pH, and now you want to bring them back to around 7.4 physiological conditions. And then when you enter cells, usually what will happen that if you go to the endosome, the pH is around 5.5, and then it will be again positive and you'll have some escape. How much escape? That's a different question.
All right, okay. All right, thank you. So it's a matter of the processing technology and lipid, right? All right.
Yep.
All right, the last question about Dan is that in the last part of your presentation, you showed the wonderful gene editing technology using Cas9 mRNA and an sgRNA. And when you deliver Cas9 by mRNA, they may have some different kinetic, which that the Cas9 should be translated. And then meantime, sgRNA might be degraded. So how do you think about the different kinetics of the Cas9 mRNA and sgRNA in cytoplasm? That is the last question.
Okay, so we have tried this and actually others as well. So we tried to bring to the cell mRNA for Cas9 and separately single guide RNA. Okay, with different kinetics, different time point. And we found that only when you put them together, for some reason, it really depends on the genes, but we have now experience with a few genes, it works perfectly. And this is really depends on many factors, mostly the gene themselves, the stability of the guide and how much time the guide is there. And now I have to do some publicity for IDT because they designed for us something that will be now commercial, but until now was not commercial. And that was really fascinating for us because it's much more stable, their single guide RNA. So probably the kinetics is that they're really inside the cells waiting for your messenger to be translated. How much time it takes the messenger to be translated? Another good question. We think it's a few hours. Basically, after 6 hours, Cas9 is already there. The protein starts to work and actually do its job very nicely. So I think that designing those single guide RNA to be not degraded for quite a lot of time inside the cells is also key.
All right. Good answers, I think. Yeah, the students may like your answer. All right, so I think that is about it. I really appreciate all the wonderful speakers of session 2. I appreciate Bruno, Randy, and Dan, and Youngbin for very thrilling discussion time. I hope we can see again, in some time, in person, in somewhere. And I appreciate all of your great contribution for this symposium. Thank you so much. I'll conclude this session. Bye.
Thank you.
Thank you so much.
Bye.
Thank you so much. Bye.
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 Thank you.